file, and then you say save a copy in Drive, and that will basically save your own personal copy in your Google Drive, and you can open that up like later on. You will actually see all the code you ran, the results you got, and if you change something, that will also be there. So let's do that. So we'll basically create a copy, open that up in a new window for you. You can actually close the original now as soon as you see that copy of 16S. And if you want, you can even rename it. So you can say Christian 16S, for instance, since it's my version, right? Okay, you open that up. You kind of see that you can connect to the runtime, clicking that connect button up here. If you do that, it basically will connect you to the cloud. It will allocate like a machine for you. Right now, that machine will have like 2 CPU cores and about like 13 gigabytes of RAM. So that will be enough for everything that we do today. And like I said, you see the table of contents. You actually also have like a search here where you could search for any text. You have some example code, and in the lower button where the folder symbol is, you actually see all the files. Right now that's empty except for some sample data that usually every Google Colab notebook has. That's not our data, it's just some general data. And we can actually start and go through that notebook a little bit and see how that works. So that's like a text cell here and kind of explains you how you can run it. We'll be running mostly command line comments here, and to kind of do that in the notebook, usually if you want to run something that's not Python code in the Google Colab notebook, you have to start it off with an exclamation mark. If you run that on your own machine in terminal, you would actually omit that initial exclamation mark because everything that you run in terminal is a comment by default. And the first thing we're going to do is we're going to go on that first cell, and you see that little play button in front of it. If you click that, it will actually execute the code. And the first thing what it will basically do is pull in all the material for today. So if you click on that, it's basically starting to create a folder called materials that contains everything we're going to do today. And that won't be taking too long since that machine has a really good connection to the internet. So basically, if you're still in that folder view, if you aren't, just click on the little folder symbol to the left here again. You can basically either update it with using that refresh button up here, but you see that the materials section is basically appearing here. And it has everything we're running today. It even has the presentations as PDF and the raw data and so on. And the last thing we will actually be doing right now is we will run the setup for Chime 2, because that's always kind of a fresh environment. Whenever you log in here, you kind of need to reinstall everything that you want to use. So in case you get disconnected from that notebook, you might have to rerun all the cells prior to that. There's a quick way to do that. Like you can click on any cell you are at, and then you can go into runtime and say, for instance, run before, which will run all of the cells, like all of the code that was before that particular step. So if you ever get disconnected today, that shouldn't be so much of a problem since we're not gonna be on that long for the first part, so that should work. So let's start that second step here, which has the second play button, and which says run material setup Chime 2 Pi, which basically will start setting up Chime 2. And you will know it kind of is doing something when you see the first output. So you see it's like downloading something and installing. That will actually take a little while. So that gives us a chance to go back to our talk and talk a little bit of what's going on here. Okay. So first of all, if that's like a bit overwhelming, you're always welcome to kind of go through that again and ask questions on the Slack, of course. Don't panic will be kind of the motto for today. Obviously, it's an online course. Things can go wrong. And we actually have a lot of contingency plans, so don't worry. Like if we're offline for a few seconds, maybe something got disconnected, but we can always get back pretty quickly and run everything again. Also, like all the output we generate is actually already in the materials folder in there as well. And it's called treasure chest because, well, it has all the treasures in there. And you will see everything that we do today. So there's always a way to get like all the intermediate outputs as well and look at them even without running anything, if that would be. Okay, but first, like maybe let's start like we're setting up Chime 2, but what is Chime, right? Like why do we actually need it? How will it help us with the amplicon sequencing data? So, so Chime is basically, it's pronounced Chime, like I'm saying it. So it's not Keim or Kime. It was created around 2010, like version 1. And it was basically came out of the Human Microbiome project that was led by Rob Knight, and Greg Caparazzo was responsible for a lot of the analysis there. And basically what they noticed is that they actually had to analyze a lot of microbiome data suddenly, and there wasn't really a good pipeline to do so. So it was really a lot of like manual work to get it to analyze, and they kind of asked themselves, can we build something that kind of makes that easier and more reproducible, where I can basically start out with the raw data and go to like analysis, and like that will kind of always work, not even for different experiments. So, but what is it really, right? So that's the official definition of CHIME. So it's a powerful, extensible, and decentralized microbiome analysis package with a focus on data processing and analysis transparency. Well, that looks very impressive. It doesn't really exactly tell us what we can do with it, but it's kind of the idea behind it, right? Like you want something that is reproducible, where it can just go from raw data to analysis. And what it actually means is quantitative insights into microbial ecology, which is what we want to get. So what is it like in a more practical term? So essentially you can understand CHIME like a set of comments to transform microbiome data into intermediate outputs and visualizations. That's what it does. So you can modify the data with analysis, and you can get to like final outputs and visualization that actually show you what's happening in the data. And it's commonly used via the command line, which is kind of how we will use it. There's other interfaces, like a web interface to it as well. You could even, there's a Python API, so you can kind of call CHIME via Python as well. But we'll be using the command line one, which is kind of the most classical one. So CHIME 2 is, in particular, was introduced in 2016, and it improves upon CHIME 1 by a lot of concepts. So basically what they observed with CHIME 1 is that it got really popular, but it also sometimes was a bit hard to see what analysis you actually makes sense or valid. So it's sometimes possible to do analysis that might not make that much sense with so the idea was more, okay, we kind of want to make it like more explicit, which outputs you can connect to what input in CHIME. And what they also want to do is like really double down on that reproducibility thing. So they have now something which is called data provenance, which means that everything that you generate with CHIME actually contains a history from how it was created, like the entire path to it. So for instance, you might start off with your raw data, you clean them up, you do some analysis. So in that final analysis output actually knows what data was generated from, like how it was cleaned up, what parameters were used for every analysis step. So you can actually take like a single output and kind of see how you got there. It has a semantic type system. So basically it understands like what data can be an input to a certain analysis, and also has an extendable plugin system. So that really opened up to the community. So you can basically write your own plugin for like a CHIME analysis, and then that can be made available for other people to use in CHIME. And it has multiple user interfaces now, but some of them are a little bit in progress still. So also, and like the really cool part of CHIME 2, in my opinion, is also that it has a really, really awesome community. So they actually include a wide range of tutorials. So for instance, we will do one today with like data that we selected, but there's a lot more on the CHIME 2 website as well, where they use like different data sets. So for instance, if you're more interested in the soil microbiome, there's a data set for that as an example there. There's also really good documentation there that really explains like all the plugins and how you run analysis. And like a really fun part of the CHIME 2 experience is the user forum, which is really an amazing web forum where you have like a lot of experts that are like super responsive, so you can ask questions. They're super friendly, and they will help you out with like whatever caveats you're finding there. So let's talk a little bit how CHIME kind of does all of that. So CHIME 2 basically generates files that are called artifacts. So an artifact is kind of like basically an intermediate data type. And those are transformed by actions. So basically, you get your initial data into an artifact, then you can do an action, and that gives you another artifact, which is kind of like a modified or analyzed version of your data. For instance, you could say that you clean up data, so you go from raw data to another artifact, which actually contains your cleaned up data. And then in the very end, you also have visualization. So visualization is a kind of an endpoint of analysis. Like it's a final part, and visualization itself cannot be an input to like another pipeline. So it's something that's actually meant for you, for human consumption, basically. So you can look at a visualization and see your result. So that's kind of what's shown on the left. So you can start with an artifact, which has like a pipeline, and the pipeline might have a method, and that can actually end up in a visualization. And to give you like an example, how that might look in practice is like here an example where you go from raw sequences, then you have kind of a demultiplexing method or action that kind of gives you demultiplexed sequences. You can like pass that on to another method that gives you like a different data type. And at some point you can always generate visualizations. And those you can actually look at. So for instance, you could get your differential abundance table, look at it. You could look at bar plots or heat maps. That's kind of the idea behind Shine. And just remember, like, Arten often represent intermediate steps, and visualizations are kind of endpoints meant for our consumption. So that's like a good rule to kind of differentiate both of them. Okay, so, okay, that's what Shine does, but what is actually amplicon sequencing? Because that was in the title, but we haven't really talked about that, right? So basically there's two large sequencing experiments that we usually do with microbiome data. So one is at 16S amplicon sequencing and one is shotgun metagenomics. And sometimes like you will see that some people say, oh, we did 16S metagenomics, which is actually not exactly correct, since actually 16S amplicon sequencing does not actually sequence the entire genome of an organism. It doesn't even sequence an entire gene. It actually only sequences a part of a gene. So the idea of the 16S amplicon sequencing is like, we're actually looking for a gene that basically every bacteria has, but it's still different enough between bacteria that I can actually say which particular bacteria it came from, what species, ideally what species or what genus at least. So, and basically what we use for that is the 16S gene. So the 16S gene is part of the ribosomal complex. And since the ribosomal complex is absolutely required for transcription, and basically every living organism on Earth does transcription, like all bacteria and all archaea have that gene. And that gene actually has a really cool structure, which we can see down there in the 16S gene, where it has those conserved regions, which are basically always the same across most of, like basically all the bacteria and archaea, and it has those interspersed variable regions that are really specific, usually for a species or a genus. So they're really specific for a particular taxon. And that makes it like super amenable to how we usually sequence something, because usually what we kind of need is we need to design a primer, like especially if we want to amplify a certain part with a polymerase chain reaction, for instance. And we kind of need that primer, which connects to something that's always there. That's where we can use the conserved part that's always in every 16S gene for every bacteria. And then this will actually amplify the variable region. And that's the stuff that we get out from sequencing that we can use to actually track back what are the organisms that we did. So basically what 16S amplicon sequencing does, it starts with the DNA extraction step. And then it amplifies like usually one or two of those variable regions. So that's what we get, and that's what we feed into the sequencer. So what we will get is actually only that small variable fraction of the 16S gene, and that's what we'll be analyzing. So that's why it's not really metagenomics, because for genomics, we want to look at several genes at once at least, or at least like a large part of the genome. And here we aren't. We're actually looking at a small fragment, which is called an amplicon, since we amplified it, of a single gene. It does have a lot of advantages, because since we're actually sequencing only a very small part, a single sequencing read, which are usually very short reads, actually covers like most of that variable region. So we get like with a single read, we can kind of sequence the entire thing. And sometimes we also have reverse reads, so we can span like a little bit larger region. We will look at that a little bit later, even though today we'll mostly be dealing with only single direction reads. So it's exactly like it's shown here. So the different approach would be shotgun metagenomics. And the idea is actually a bit simpler. So in that sense, we basically chop up the entire genome of the bacteria, and we try to cover it with as many reads as we can to kind of get back to the entire genome. So if all of the reads in theory would overlap, we would be able to kind of get back the full genome. In most cases, we don't, except for like very abundant things, but that's kind of the main idea. But the thing is, we obviously need way more reads to cover the entire genome than we need to kind of sequence that little region well. So there's a kind of a trade-off here. 16S amplicon sequencing can like kind of give you good results, even with low depths. For shotgun metagenomics, you need to generate a lot of reads, and except that you need pretty good sequencing depths. But the base idea is kind of similar in that we start out with DNA extraction, but then we actually do a fragmentation part where we chop up the genome into a lot of small parts. We ligate adapters to that. So that's kind of like the PCR, but it's not really an amplification. We actually just glue an adapter to it that will kind of make it stick to the sequencer, and then we're doing the sequencing. So that's the two approaches that there are. The second part of the course can actually use both of those as input, but we'll be using the part on the left, which is 16S amplicon sequencing. So what is the data set we're actually going to look at? So what we are going to look at is data from like one of the kind of very dangerous pathogens that we can encounter in a hospital those days. And we'll be looking at 16S amplicon sequencing data of the V4 region from human fecal samples. So usually what we get like those DNA samples from is that we kind of take a poop sample and then we extract the DNA from there. Poop actually contains a lot of bacteria, so it's like really good for that. Every time you poop out at least 200 grams of bacteria. So we can sequence those. There's a lot of material in there that can be sequenced. And we're looking at the V4 region. So the 16S gene, like I said, has several variable regions. They are called, for instance, V1, V2, VC, V4. And for bacteria, V4 is one of the most informative ones. So it gives like the best taxonomic separation between them. We'll be looking at 8 samples in total for healthy donors and for individuals with recurrent infection. And I put the link here for the original publication that was generated by Mike Sadowski. And Mike Sadowski is still a major player in fecal microbial transplants, where you kind of can, which is still one of the best cures we have right now for C. diff infections. But what actually is a C. diff infection and why is it so bad for you? Let's look at that. So we have that great visualization here, which was generated by Stephanie Swegler, which was a high school intern at the Institute for Systems Biology. So often we are treated with antibiotics. That just happens. You might be a little bit sick. You might have an infection. Antibiotics are pretty common in our setting. The problem with that is that our gut harbors a lot of good bacteria that help us to stay healthy and also to help us to like kind of digest the foods that we can actually not break down ourselves. So we kind of will attack those as well. That just happens. Actually, most of the antibiotics, at least if you take them, if you eat them up and swallow them, will actually get to your gut first, and will take a while to actually get to your bloodstream. So you get really high antibiotic concentrations in your gut if you take antibiotics as a pill. So basically, if you take antibiotics, what you often do is you eradicate large parts of your gut microbiome, which is kind of between the A and B step here. So you get kind of that state where a lot of the bacteria in your gut are dead by now. And there's a pathogen called Clostridium difficile, which really likes that stage. So it can actually detect when like the gut microbiome load or the microbial load in your gut gets really low. And as soon as it detects that this happens, it becomes like pretty aggressive. So it gives you like a massive infection, which is not only the worst diarrhea you will ever experience in your life, but it can actually like it's a really painful experience, and it can definitely kill you if it gets really bad. How does it get really bad? Well, usually what happens is if you get the C. diff infection, what we do is we give you more antibiotics. Like we give you an antibiotic to kill off C. diff. And in some cases that kind of works. So you get back to a state where, well, you still have a kind of a damaged microbiome, but maybe you can recover it. But in some of the cases, what happens is actually that the antibiotics are not successful in killing off C. diff. C. diff actually doesn't really respond to it. You actually eliminate even the little bit of the remaining healthy microbiomes that you had, and you get into what is called a recurrent infection cycle, where basically you will get antibiotics, you might feel a little bit better for a while, but then actually you get a huge outbreak of infection again at some point, and then you take antibiotics again, and that goes back and forth, and you get into a really unhealthy state where you just feel miserable because whatever you get treated, you feel a little bit better and then get worse over time. So what we have today actually and what works is that we use a fecal microbial transplant for that. Like that actually works for recurrent infections. And that paper, what we're looking at was one of the first actually looked at like how that might be and under what like circumstance that actually works out. What we can take is we can take like a super donor, and that that's a really healthy person with like an excellent gut microbiome, like it's a really good one. And we basically take super poop from that person, which kind of provides us with like a microbiome community that is very resistant and very strong. And if we give that to like an individual which has that recurrent infection, that actually outcompetes C. diff. So it kind of pushes C. diff out of that system. So you get like small amounts of like Clostridium difficile and the healthy microbiome. And at some point, C. diff can actually not compete anymore and gets eradicated in that system, and the person is healthy again. And that's right now, it has kind of like an emergency approbation by the FDA, and it's used, and it's kind of, for people that have recurrent infection, that can be almost like a miracle treatment. Like within like a few days, you suddenly feel great after having felt like miserable for maybe a year or something. So it's like a kind of a really cool thing that works right now. So we'll be looking at individuals that are either like healthy, like the outstep, or we're looking at individuals that have the recurrent infection of C. diff. The interesting part is here that those individuals don't necessarily have really high Clostridium difficile abundances in their gut microbiome, because like we said, they might actually just be in a state where they're kind of feeling okay, but we know that those individuals will be going back to those infection cycles. So they might actually feel really bad 2 weeks from now. So like one question we can ask is, well, it's like, if we look at the gut microbiome composition, do they look different, for instance, from like healthy persons? And can we find like a signal that makes it like can we find the underlying like cause why that particular microbiome is so bad of keeping C. diff outside of the system? Okay, so what will we do today? So we're starting from raw sequencing data. So basically just the sequences of those small reads that we had. We will kind of clean them up, trim and filter. Then we will do something which is called infer sequencing variants, because sequences do a lot of errors. They have gotten better, but still, like, a sequencer will not give you the exact DNA sequence of whatever you put into it. It will give you a lot of DNA sequences which have like small amounts of errors in the sequencing. So what we actually want to do is to get back and say, okay, what is the original DNA sequence that were in our sample, even if our sequencer will introduce errors into them. That's what this will do. Then we will kind of try to check who's actually at the party. So who are the bacteria that we have present in the sample, which will talk a little bit about normalization, and we'll do some analysis of ecological diversity and also disease relevance. So let's kind of look at what we actually get from a sequencer, right? So basically, after you have done your experiment, you extract the DNA, you did all your library prep, which basically library prep means doing all of that, what is necessary to make a sequence actually be sequenceable by a sequencer. So you kind of have to chop it up, you have to add an adapter to it so it actually sticks to the sequencer, and the sequencer knows where to start sequencing. And what you often get is like those really weird files, which are called FASTQ files, and often they're also like compressed, so they're like gzipped. It's called, so they are kind of made a little bit smaller, more efficient to store, and they have like often those really weird names. So for instance, if you use an Illumina sequencer, and most days we usually analyze them with base space, you kind of get a file name like that looks kind of odd, but actually it has some logic to it. So basically the first part will be your sample ID. So often that should be something that you gave to the sequencing unit, and it will kind of have a second part of it, which is like kind of the order. And that's usually the order was injected onto the sequencer. So you kind of know which samples were processed first and which ones were processed last. Then we have also run or lane sample set. So usually on a single sequencing run or a single sequencing experiment, you can only kind of sequence a certain amount of DNA before you kind of saturate in the sequencer. A lot of sequencers, to alleviate with that, those days actually have several lanes. So it's basically, you can kind of imagine that like a highway. Each highway is like a sequencing experiment, and you can have like 4 lanes in parallel, and that gets you to push more data through it. So that will tell you on what lane that was sequenced, or if you actually did like a second sequencing run. And then the last part will tell you the redirection. So you can actually go forward, and you can also go reverse. So you could basically either get 2 reads for each DNA sequence that you put in, or only 1, like a forward read or a reverse read. And then the last part, the 001, actually means nothing. And how kind of, what does a FASTQ file look like? Well, it's actually fairly simple. So it starts out with a part that starts with an @ and that actually only tells you kind of some information about the actual sequence. So you kind of start out with a sample ID. You get some weird number. Usually that's kind of the read number. So you generate a lot of reads for a single sample, right? Like there can be millions if you want to. So it kind of tells you which of the read it is. And it will also tell you how long the read is, because depending on the sequencer, those lengths that can have different lengths. For instance, we started out with really short reads, like with Illumina, like the first sequencer would probably generate like 60 to 80 base pairs. By now, today, we can easily do 100, 150. And if you use a MySeq, you actually get like 250 and something around that. And that's actually like not really a completely deterministic process. So sometimes, like, since it's still like a polymerase that kind of synthesizes the read, like sometimes it starts a little bit earlier or jumps off like a bit earlier from the sequence, so you might get like a shorter or longer one. So, and the second line is actually the pure DNA sequence that you got for that read. So that's the actual sequence that the sequencer gave you, but it might have errors, right? So to kind of be able to judge which of those bases has high confidence, which not, the sequencer actually has kind of a way of telling you, okay, I'm really sure that the first letter, for instance, is a T. And it does that by an additional line, and that gets separated by a plus sign, which gives us the quality. And that looks really wonky and weird. And actually what they did is basically they said, okay, we have to encode the quality of that base. Obviously, let's say I would do that with a value, right? I have qualities from 1 to 40. If I have a 1 here, I could put a 1 here. That's fine. But if I put a 40 here, that would be a bit weird, because now I don't know, is it like a 40 for the T, or is it a 4 for the T and a 0 for the second base here, which is an A? So they have to be like one letter symbols to encode the quality. So what they did is they basically took like all the letters and numbers and a lot of different weird symbols like question mark and at, and each one of that like encodes a particular quality. So usually we have quality scores starting from 0 to 40. And that kind of gives us like an idea how much confidence we have. So 0 would be basically that's a garbage sequencing base here, so we have no confidence. 40 would be extremely good. As an intuition, because it's a logarithmic scale, so it's a bit weird to understand, but a good rule of thumb is a quality of 10 means 10% error. So that's pretty easy, but then it goes exponentially better from there. So a score of 20 actually means you're 10 times better. So you only do 1% of an error per base. A score of 30 would be 0.1% of error. So basically we say 20, which is like 1% of error, that's pretty good. Like, and usually we kind of want to stay above 20 more or less in the qualities here. Yeah, it's kind of sad that this format was like invented like a while ago. If it were invented today, we would probably have an emoji quality code, which would be much funnier to have like a smiley face for good quality and angry face for bad one. But yeah, that standard wasn't invented until later. Okay. Cool. So we have our real raw sequencing data, but QIIME2 only operates on artifacts, right? Like actually QIIME can only work from artifacts. So how do we convert our data into an artifact? It's a bit of a chicken of an egg problem, right? Like we need an artifact from our original data. And so how do we get there to actually feed it in there? So let's look at that. Let's go back to our notebook and let's actually check if we're good here. Let's move that around. Okay. So hopefully all of you see something like that. So actually it tells us that it did quite a lot of stuff. So it installed QIIME2. And in the very end, it should tell us everything is okay. So you can actually start using QIIME2 now. So that's good. So now we have the installation done. The installation might be a bit easier if you do it on your own machine. Actually, we kind of have to do some hacky things to make it work in Google Colabs. That's why it has a few steps here. And what we will do is we'll also run like the third part here. And that basically only changes our working directory to the materials folder where we have our raw data. So if we look here in data, for instance, you actually see we have those FASTQ files that I talked about. And they are named with like a weird sequencing thing here. Basically, that's the idea that we have in the sequence read archive, which is like a huge repository where you can actually deposit your raw sequencing data when you publish it. So that's what those samples mean. And we have an extra file here that will tell us what each sample means, but we'll look at that later. So, okay, we changed to the materials folder. And here again is what we will do today. So just to get back. So what I said is we kind of have to start importing the data somehow to get an artifact. And that's the first thing we will do. And that's also the first chime comment we will run. So let's look a little bit how the chime comment looks like. So often what we get is you get like, you always start every chime comment with chime. And again, like the exclamation mark here means basically, okay, we'll be running a comment in a comment line and not Python code, because the Google Colab notebook actually defaults to Python, as we will see in the second part as well. So usually you just start with chime. Then you kind of usually name the plugin. So that's like the sub part of chime that you're using. And we were using the tools part. And then you kind of have the method or the action, what you execute, and that will be import. So basically we're telling them, okay, chime, look in your kind of tools selection and use the import method. And now actually tell you what to import and how. And the first thing is a bit odd. So we say it's a type and we say, oh, it's sample data, sequences with quality. And like I said, like chime has now this way of checking that actually the data you give it for a particular analysis makes sense to it. And that is done with types. So what we're telling them right now is that we actually have samples, and those are sequencing data and they have quality scores. So it kind of expects FASTQ files actually. Then what we have to provide is like a manifest file. And that manifest file is actually really simple. Let's see, we can open one here maybe. So if you ever want to download a file, and I will show you that again in a few minutes, you can go again in that folder symbol. I'll just do that from the start. You go in the materials folder, open that up. And then, for instance, you have that manifest CSV file. You can click on the 3 little dots next to it and you can say download. It will actually download the file. It did it here for me, and I can open that up, for instance. Or you might not actually see that, but that's how you can look at it if you wanted to. And it's a very simple file, actually. It actually only has a sample ID and then tells you where the path is where you can find that file. So in our case, if you download this, like if you want to download it, again, it's in the folder symbol, materials, and then the metadata TSV. You kind of, if you hover over that, you actually see those 3 little dots to the side where you can download it. You don't have to do that right now. It just, we'll show you again how to do that in a second. But basically, it's kind of describes your sequencing files. It tells us, okay, that sample is in that FASTQ file. And if you're forward and reverse files, you can actually look at both of them. And yeah, exactly. You can also look at it in the notebook if you wanted to. So, but it's, yeah, basically that sets up the files. Then we kind of have to tell you what is the artifact, the chime artifact called, where you actually want to save that. So in this case, we will just call it cdiff.qza. Usually chime artifacts always end in QZA, whereas chime visualizations end in QZV. So the Q is for chime. Z actually means zipped or compressed, because actually all chime files are just zip files with a different ending, and the A means an artifact. So cdiff.qza will our output file them. And then we actually have to also define, okay, what do we actually have as a data file? So one is the chime type it will be imported as, and one is the actual file type of your data. And here we are really specific and we tell them, okay, we have single end files. It's FASTQ. We're giving you a manifest so you know where to find the files. And the last one is basically the standard that's used for the quality data. It's almost always in those days going to be exactly the one here. It looks really weird, but basically when that sequencing was kind of the FASTQ files were kind of introduced, like initially, like there were different standards on how to encode quality scores. For instance, one only went up to, I think, to the 20s or something, one went higher. So there's like different versions to it. And that's kind of denoted by that thread here, which is kind of the standard of the quality score, but you can basically always do that. Yeah, and the exclamation mark does not comment out the line, but the exclamation mark just tells us that it's like a comment that should be run on the comment line. You can actually, if you run that on your own equipment, you would ignore that exclamation mark. It's not commenting it, it's just telling the Colab notebook that it should be run in the terminal or the comment line, basically. Okay, so we can run that. That will basically take all the data and put it into a single artifact and save it. And that artifact, and it tells us here, it says, okay, I imported it to this particular file. And actually, if you would update here, you would even see that it's now there. So that's our artifact now. So we kind of got around that chicken and egg problem and actually imported data now. So that's pretty good. Okay. And yeah, maybe just to reiterate like how that comment looks like. So we always have that same kind of structure here. So it says chime, plug in the action, and then we have something like this dash dash I argument, O argument, and there's kind of a rule to that. So if it starts with dash dash I, it actually means those should be your input files. If it starts with dash dash O, that's the output files, so the artifact you want to generate. You also have dash dash P, which are parameters. So you can kind of say, oh, run that analysis, but with that particular settings. And you have dash dash M, which is metadata. Metadata means like extra information about your samples. For instance, right now, you notice we imported the data, but we didn't tell Chime at all like which parts of the data are, for instance, healthy individuals, which ones are sick individuals. But at some point in the analysis, we might want to incorporate that information. So that's like kind of how we can build that in. So that's like the general structure. And the names of those are specific to the plugin. And if you look at the documentation, you can see it. Right now, what we probably want to do is actually look at the sequencing qualities, right? Like we sequenced, we don't know, like if that was done well or not, if it worked out nicely or not. So let's kind of have a look at that and go through it a little bit. The first thing we can do is we can use the demux part of Chime 2. So demux actually means demultiplexing. And the idea is there that usually, like even though we have several samples, we often sequence them together into a single run on the sequencer. And the way we actually get back to individual samples is that we like actually add a little adapter to it that has a barcode. So basically we, before the actual sequence or the DNA parts that we're amplifying, we're actually adding a little barcode, which is usually like 8 bases to 10 bases in some cases long, that kind of tells us from which sample it comes. So every single read from that particular sample will start with the same sequence. In our case, we actually already had our data separated by sample. And actually that step going from the raw sequencing data to that kind of per sample separated data is called demultiplexing. In those days, if you use Illumina and BaseSpace, for instance, but also with other tools, like if you use an Oxford Nanopore, whatever you use for sequencing, actually already does that for you in most cases. So when we started out with 16S sequencing, that wasn't really the case. So Chime 2 still can do that. It can demultiplex if you have everything into a single FASTQ file and like all of the samples are kind of together there, and you have some barcodes at the end, you can use it. But we don't need to do it. But it has that extra action, which is called summarize. And that is really cool because it will give us a visualization of the qualities, like a visual representation where we can just check how well the sequencing worked. So we will use that part of it. So if you ran this previous part, so you should see that green text here that it was imported, you can run this part as well. And that one will basically analyze the qualities and will end up in a visualization. So in theory, that's something we should be able to see. But well, I'm not a computer, so I don't know how to read that weird QZV thing. So let's see how we can actually open that. And the trick how to open is, and like maybe the easiest for today is kind of, there's a tool which is called view.chime2.org. It's an online tool that will actually open the visualization and show it to you in your browser. You can look at that. So if you click on that link, it opens that up in a new window. You kind of see that thing where it says drag and drop, or click here. Okay, if you want to drag and drop here, we actually have to download it first, right? So let's actually do that. So we actually don't see it here yet. We might have to, again, go on the refresh button here to actually see the file that was generated. We see the qualities.qzv file. Again, that's in the materials folder. We go on it, click on the 3 little dots, go on the download one. That one will download that file. You can say, oops, download the wrong one? I'm sorry. It's the qualities one. Okay, now I downloaded it in my case. And now we can kind of drag and drop it here. So select it, for instance, from our downloads. Wait, where did it save it? Here it is, qualities QZV. So wherever your download folder is. So if you open that up, you get something like this. So first, it will kind of tell you like an overview of your sequencing experiment. It tells us like, for instance, how many forward reads we actually have per sample. And we see that in the sample with the least amount of reads, we have still 19,000 reads. Like a kind of good rule of thumb for 16S amplicon sequencing is you want kind of around like more than 10,000 reads per sample. You can do with a few thousand. You definitely never want to see less than 1,000. That will not be enough to actually do a good analysis. But we see that actually we're pretty good. So we have at least like almost 20,000 reads per sample. Actually, our median and mean are like 60,000 reads. And that's a good like density to kind of get most of the bacteria that are in there. Okay, and tells you like just in a histogram. The most interesting part here is basically going on the interactive quality plot. So if you look at that, you actually see for each position of the read, so that read was 150 base pairs long, apparently, we kind of see the distribution of qualities in kind of a little part. And you can actually even select it and lock it, kind of zoom into parts. So if you have that little crosshair, you can go to a certain region. And then if you double click, it goes back. So kind of what we said is we want to be above 20 more or less, right? So if we look at that sequencing data here, so we kind of see the distribution of qualities for each position of the read. Like if you had to clean that up, and basically cleaning up, what we often do is we kind of say we cut it, for instance. So for instance, we could say, hey, let's remove the last 10 bases from it and actually only take the first 150 bases of that read. So if you look at that, what's like your opinion, what we should do? Should we cut it maybe at position 50, or should we cut it or leave all of it? What do you think is a good idea here? You can post that in the Slack. And you can also see that basically all of our reads were 151 nucleotides long, which is actually, it's like 150 base per. Yep. Yeah, a lot of people say that it looks like the first few bases aren't that great. And then at the end, there's definitely a drop here, right? Actually, if you have longer reads, like 250 base per reads, what you often see is that around like 200 base pairs actually drops quite drastically. It can even go lower than 10. That's quite common with like longer reads. With shorter reads like that, you usually see they look pretty good, but the very end might look a bit dirtier. So here it's kind of dropping below the 20. It's not really bad, like it's not getting to the 10 yet. And in the beginning, that's actually interesting. So that can either be really mean, you have a little bit lower quality, but also like kind of what the methods that Illumina uses to tell you the quality of each read is actually performing worse at the beginning of the read, because it usually needs like some bases before that. So it actually less confidence here that what it actually reports as quality is correct. But yeah, I think, yeah, cut only the last nucleotide or cut only like the last few would probably be a good option. And if you want to be really conservative, you could cut off the first few ones. So let's look back to the notebook and let's actually try to do that. So that decision what to cut off the read is actually something that we have to do. So so now where we have made a decision, we can actually implement it. And that will be one of the steps for actually a really large analysis step that's run into a single comment in Chime, and that we'll be following now. And that will be a step which is called denoising. And we will talk a little bit what that is, but we might actually start it off earlier to kind of yeah, have some ideas. So what's happening, and no, to be able to wait until it's finished, basically. So let's see. So the next step will actually do a lot of stuff. It will filter and trim the reads. We talked about that a little bit. Filtering basically means is that even though we have that overall quality, we see there's a distribution to it, right? So there might actually be like a really bad read here that has like really low quality throughout. And there's a way, like basically there's a default value, and usually that's usually around that like 20 to 10 value, but will actually throw out that single read that's really low quality. But then we also want to usually do that truncating where we cut off like a particular part of all the reads. So that's basically what happens implicitly in here. So we run that comment by saying chime. That one is the data2 package, and we will talk about what data2 is and what it means. And it has a denoise single. So basically we say single because we have only single read data. We only have forward reads, no reverse reads. So we have our input data. So we needed an artifact to start as the input data, and we do have that. So that's good. And we will truncate the length at 150. But actually we said we don't, right? We actually want to truncate the length maybe at 140. So you can do that in here if you want. So we can do, say, for instance, truncate the length. So in SVC, that's actually a parameter, right? So the input artifact, that's an input. So it starts with an I, and then demultiplex sequences. They are demultiplexed because they're by samples. And we will for now maybe only cut off like a part of it. There is a parameter in here, and you can look at the QIIME documentation if you want to find out what that is called, to actually also cut off the first few nucleotides if you want to do that. For now, we will cut maybe down to 140 if you want. And we will actually, so that generates a lot of different outputs. We could actually give file names for every single one. A shorter way would be just to say, oh, like just save it into a single directory called data2, which is like kind of the name of that plugin that does it. And we will also use the verbose action that will actually tell us, and so we can see how it's running through the steps. So let's start that. That will actually take a while because those data have quite a lot of reads. We don't have that much computational power here. So while that is starting to run, it's actually running R here. So we can actually understand what's going on. So like we said, we brought in the big guns here. We started with the data2 steps that does a lot of things at once. So we said we filter and trim the reads. So we'll throw out reads that are really low quality. We'll also cut them at like 150 base pairs. It will do something which is called finds the most likely original sequence in the sample. Like I said, sequencing makes errors. So what are the actual sequences that we put in there? It will remove chimeras. We will understand what that is in a little bit. And it will actually count the abundances and will tell us for every unique sequence in there, what is the abundances. If you can't run QIIME2, you might not have run the initial setup step in the top of the notebook. So we talked about pre-processing. Like we said, we can trim low quality region. It actually does a little bit more than that. And we don't need to specify any parameters. It just does that automatically. It removes reads with low average quality, like I said. It also removes reads with ambiguous bases. So sometimes the sequencer might actually say, I can't even tell what base is on that position, and just puts in an N. And N is obviously not a real base in DNA sequence, and it just kind of means that I don't know what that is. And also removes phi X. So basically, the 16S gene, like I said, is very conserved. So basically, we're sequencing exactly the same thing in every sample with some differences in the variable region. The problem is that sequences are often really bad if every single thing is exactly the same. And then to kind of alleviate with that, we actually like add in like an additional sequence, which is phi X. That's like a virus that is really well known. And that's actually added to the sequencing run to make like a bit more heterogeneous sample or more diverse sample. And usually that increases like the sequencing performance quite a bit. But we need to remove those. And that's also happening in here if you have that in your sequencing run. Now let's look a little bit about those weird alternative sequence variants. So like I said, what actually happens is, let's say we only have 2 unique DNA sequences in our sample. One is like the blue one and one might be the black one. What happens is the sequencer makes errors. So it might actually not give us exactly those reads, but it will actually give us reads which have like, for instance, different one position of the read. So where we have only one sequence, we get 3 different reads here. And for another sequence, we get another 3 different reads here. So we can't just take out like the read and say, oh, that's exactly the sequence that was in there. So if I see like 1,000 different sequences, I would assume there's 1,000 different bacteria in there. That's like, or species in there. That's not true. Usually it's way less because the sequencer does errors, so we inflate that number a lot. What data2 does is actually, it tries to get those sequences. The problem is to get those sequences, what I really needed to know beforehand is kind of, if the sequencer tells us I have like a quality of 10, what's the actual probability of making an error like here? And even though Illumina kind of tells you 10 is like 10%, that's often not really true. It's not the exact value or the probability of making an error in that base. Also, it depends, right? Like it tells you 10% error, but maybe it doesn't make an error if the actual base is an A. Like it always gets that sequence perfectly, but if the base is a G, it might be really bad, right? And it makes like a higher error. In average, it makes maybe 10% error, but for each base, that's different. So data2 also has to estimate that. So again, we kind of have a chicken and egg problem. Like to get back to the original sequence, I need that error profile, that error probability set. But to get the error probability set, I would actually need to know what the original sequences were. Then I could just look and see, oh, like all the reads from that sequence, like how many errors did they make in each position. So whenever we have that kind of chicken and egg problem, like as a computational biologist, there's like kind of a blanket solution, which is called expectation maximization algorithm, which actually is able to solve both at the same time. That's the basic trick here. And we don't need to understand the exact details of it. What we need to know is that it can actually do both. So it can find the error profile, so it can tell you, oh, like what's the actual probability that an A gets substituted for a T, for instance, if you have a particular quality score. And it can also identify which are the original sequences in there. And it does both of that. And that's really cool. And when that came out, it just blew most of the other methods that existed before and out of the water completely. So they did mock experiments where actually they knew exactly which bacteria they put in there. Like for instance, they would put in 10 strains and DADA2 would tell them there's exactly 10 unique sequences in there. So it was really good at like getting back to that. So most of the time what we get out from DADA2 is like kind of the variants that we actually had in the 16S gene, and those are called sequence variants or alternative sequence variants that tell us like kind of what is the most likely actual 16S variable regions that we had in our sample. But there's actually another little problem in there. So like we said, we do amplicon sequence analysis. So, oh yeah, amplicon sequence variant, sorry, not alternative. If we do that, we actually amplify a part of it, right? And there's a little issue here that what we're amplifying is extremely similar. So basically, even though it's a variable region of the 16S gene, there's still most of that sequence is kind of exactly the same across all of them. So PCR, kind of how it works is what is shown here. You start with a primer. The primer usually get extended and then kind of gets you the entire read based on the template that you had in your sample. The thing is here, sometimes what happens is that like the polymerase is not 100% accurate. Sometimes it just kind of aborts earlier. It falls off basically of the template strand. And it leaves us with like a shorter sequence part. And now there's a problem here because that short part might be very similar to a lot of different DNA sequences that we actually have in the sample. So it might actually align and connect with one, hybridize with like another strand that is actually not the same one. It's not the green sequence here. It's a different one. It's just very similar. And then like a new polymerase gets on there and kind of continue sequencing it. And then we get something what is called a chimera, or more exactly a bimera in this case. And this is like a bit complicated because it gives us like a weird intermediate sequence. It's actually not like from a single DNA sequence in our samples, we actually get like a combination of two. And that's kind of not perfect, right? Like this will really screw up our analysis because if one comes from one bacteria and one comes from the other, I now suddenly see something that is like half and half, like a Franken bacteria. So we won't know what that is. And also like, maybe as an additional question, since we kind of, the primers usually like target a particular region of the 16S gene and maybe like to kind of refresh what we saw before. So usually you have two primers, you have one in the beginning and one at the end of the sequence. So in this case, they started at position 515 and the reverse primer in 806. So how long is actually the entire fragment that we're amplifying? We can think about that a little bit. So let's kind of check out where we're at here. Okay, that looks really good. So kind of just to check that DADA2 did what we expected it to do, etc. It was filtering, which is kind of the cleaning step. And then you see that it first actually learned the error rates. So it kind of used a lot of different bases from all the reads to learn the exact error model, and then actually denoised the sample. Denoising means it removed reads that seemed to be just like a version of the original reads that was in the sample. So now we only have those amplicon sequence variants, which kind of tell us what is supposedly the most likely actual distribution of variable regions that we had in our data. And then it removed chimeras. It used a consensus method, which basically means it checks across different samples. And if it sees the same thing as a chimera in several, it gets removed. And it wrote the output. So we got actually three things here. We get a table that basically tells you for each amplicon sequence variant, in each sample, what is the number of reads that we had from that particular variant. So that can be like 100 reads, only 1 or 0 as well. If it wasn't in the sample, it wasn't detected. We get like a feature data sequence table, and that actually just contains the actual sequences that it thinks were actually in the sample. So that's already removed all those error reads basically. And we get some denoising stats, which is like an artifact which we could analyze later on to kind of look at it. So that's that's the data. It's all artifacts, so we can't really look at it right now. Like we actually need to do something else to analyze it. And in case this didn't work for you, or you like joined a little bit later, you can also run that. Don't run that right now if it worked for you. If you see green text here, don't run that cell. But if you for some reason like don't want to wait for that to finish or didn't finish, then you can use that, and we'll use like the cached version. Okay. So why did I ask you how long the amplified fragment is? Because that's something you should definitely ask yourself when you design the sequencing experiment. And why is that? Well, because it can happen that you get like a really weird performance here. So basically what can happen is that you see that the table is essentially empty. You generate no data from it, for instance. And if that happens, it's usually because you usually truncated too much of the read. So for instance, if you have for paired NC data, it can often happen that you start out with a well-designed experiment. So that's your V4 region, for instance. You had a forward read, you have a reverse read. They actually overlap, and that's what you ideally want. Like if you really want to sequence the entire region, you kind of need those reads to overlap. Well, that's all good. So you see, oh, my experiment is well designed. I do very aggressive like quality filtering because I want to be really stringent. So I cut off a lot of that read. And then it can happen that in the step where data 2 actually tries to merge those to get the entire V4 region, that we actually get no overhang here. And that would be bad. So basically here we are having some problems. And might actually like give you some error that doesn't work. It needs at least 12 bases usually between forward and reverse reads by default. And you can calculate that. So if your amplified region is 450 base pairs long and you have 250 base pairs reads, that might actually work, right? Because you cover up to 500. But then if you trim the last 30 base of each read, you would truncate the lengths to 220 each. So your entire fragment is 440, and that leaves no overlap for the 450. So this is something you should actually kind of check. You can also adjust the minimum overlap, but usually like 12 is actually pretty good. So I wouldn't go much lower than that, because then you get like mergers that don't work. Sometimes can also happen that you get like a lot of the reads lost as chimeric reads. And that might be like, that can be a problem with the PCR that you run in the experiment. So it can be really an experimental issue. It can also mean that you actually did lower that parameter too low, and it like kind of merged stuff that shouldn't have been merged in that step. However, like I said, that's the 60 variable regions are very similar. We do expect that we always have some chimeras in there. And usually losing between 5 to 25% of your reads to chimeras is normal and does not require any adjustment. That just happens. It can sometimes even go up to 40%. The important part is to check how many reads do I have left, like that 10,000 read goal. If I can still hit that afterwards, that would be good. So actually, let's have a look and see how well we did in that analysis. What we can do is basically, we can use a tool called Tabulate in QIIME that takes those denoising statistics that we got, which is just an artifact, and turns it into visualization. So that table is somewhere saved in there and will actually return into something we can look at. So if we run that comment, we can actually download it again and look at it again in the visualization. Okay, so now we get those data 2 denoising stats. So that's in the data 2 folder. So I open that up, and you see that we have 2 versions of it now. And one is ends in QZV. That's the visualization. That's what you want to download if you actually want to look at it. So we can download that one again. Can go back maybe to the main page of QIIME2 view. So that's view.qiime2.org if you want to open that up anew in your browser. And again, I click on it. I choose the denoising stats this time. We can look at that. Okay, so now we kind of see for each sample, how many reads did we have initially, how many were quality filtered out, and actually that looks fairly good. So we see that about 99% almost of the input filtered, like past the filter. So only 1% were removed as either like having low quality. So that's pretty good. Then we did the denoising, and we still captured most of it. So it worked pretty well. And then we kind of see that really like kind of expected drop in the chimera removal, right? Because like a lot of those reads are kind of came from chimeras. So we kind of don't want to use them because they might be really misleading in terms, especially if we do taxonomy assignments later on. So here we see, but we're still doing pretty good. Like here we go, for instance, we go from 87,000 reads to like 70,000. That's still far above the 10,000. And even in the worst sample, we got from 19,000 to 17,000. And we see, like I said, 5 to 25%. Here it looks like an average, maybe 20% got lost. So that's pretty decent. And we're pretty much still left with like a denoise data set that's still pretty usable for us. Okay, I'll leave that. Cool. Yeah, now we actually have data, so we can actually start and do some analysis with it. And let's maybe get back to the presentation here and ask ourselves. So now we have that table that contains the counts for each amplicon sequence in each sample. We also have a list of those. So what could we do with these two data sets? What quantities might we be interested in? And one thing we can do, even if we don't really know like what exact bacteria those came from, we can kind of look at it like, oh, how many different ones are there? Like, are there differences between samples already in that distribution of those sequence variants, or are they all the same? So that's like a question we can have. So what we're going to start looking at is diversity metrics. So we're usually interested in two different families of diversity metrics. One is alpha diversity, and that's like the diversity within a single sample. So you take a single sample and you ask, okay, how diverse is that in an ecological sense? And the other one is beta diversity, and that means ecological difference between samples. And we have kind of different steps here. But before we actually do and like look at it again, let's do the same what we did before, go back to our notebook and kind of start with the first parts. So we'll actually kind of explain what all of that does, but we will start by constructing a tree here. So you can start playing that already. And then we will do like a diversity analysis in QIIME 2 that will actually allow us to generate like a lot of different metrics. And let's start this as well. We can actually schedule it by just clicking play. Even if the first one is still running, you can click play on the next step as well. And it will basically like run both of them in succession. So that's like kind of perfect to wait until it's done. It's actually pretty fast, but just to be sure. So again, like I said, alpha diversity, how diverse is a single sample? So, and if we kind of like maybe impiction like different bacteria here with like rectangles and triangles and circles, like the first one intuitively would say, okay, there's a lot of different things in there, so it looks kind of diverse. But it can happen that we kind of have the same different bacteria in there. But yeah, like one is still dominating. So that's like somewhat less diverse than that. We can have a very non-diverse sample where everything is basically the same bacteria in here. So we see there's like different nuances to it. Like the first one is richness. How many taxa do we observe? Like that's usually called richness, like number of observed taxons, for instance, or a Simpson index, which like adjusts that a little bit for like certain errors you can do. So that would be like the most obvious thing. Like you just look how many different amplicon sequencing variants do we have in each sample. But then, like I said, like between the first two steps, like just having the same number of different ASVs can still have like kind of a difference. Like it can still be dominated by one. And we have like very few reads from, for instance, two different amplicon sequence variants. And that's kind of captured by evenness. The abundance is distributed across taxa in a single sample. And that would be the evenness index. And in most cases, we actually want to do like an intermediate between the two, which is called the Shannon index. The Shannon index actually combines both richness and evenness. Actually uses like a physical concept, which is related to entropy. Like you could kind of like an intuitive interpretation of the Shannon index, because the formula looks kind of weird, has a logarithm in there and does some odd stuff. But the intuitive interpretation is, if I give you a bag of your sample, like a magic bag, you kind of pull out the first bacteria and you say, oh, wow, that's Clostridium. And you grab in there well, again, like how surprised will you be of the output? Like for instance, let's say you grab in there, it's Clostridium, grab in there again, Clostridium, grab in there again, Clostridium. You're not really surprised the third time, right? You're like, okay, everything was Clostridium. But now you might like have a different setting, you grab in, get Clostridium, next one, Bacteroidetes, next one, Firmicutes. If I would ask you, oh, what's the first one I will pull out here? You're like, oh, well, I won't know, right? Could be anything. So it would be very surprising. So that's kind of like what the Shannon index quantifies. Like how surprised are you if you like randomly take out bacteria from that sample? And that combines like the richness and the evenness into a single score. So it's kind of a really good measure for that. And if you actually want to kind of do statistical testing, for instance, you could check for all the samples that are healthy and all the samples that are sick, is there different alpha diversity? You can actually do that with pretty common things. Since you have one value for each sample, you can use a t-test or Mann-Whitney U-test if it was something non-parametric. So it's like classic univariate testing. So we actually don't have to depth that much here. It's a single value for each sample. So it's like a good indicator, like if you want like a global descriptor of what each sample looks like. In terms of beta diversity, we could ask like how different are two or more samples from one another. So that's a bit different. So for instance, you could have like sample 1 and sample 2. In that case, you kind of see, well, like both of those samples, like they contain pretty much the same taxa, right? And the abundances, well, they're not exactly the same, but it's kind of very similar. But you could also imagine a setting where like in sample 1 and sample 2, they don't even share like a single taxon, right? So they're very different. So, and that's again, like the two nuances here. One is like the unweighted, like how many taxons are just shared between samples. In that case, you would say the first two samples are actually the same because they contain the same taxa, right? And so the second two set here would actually be very different because they don't share a single taxa. But you can also do like weighted, and then you don't only look at if they share the same taxa, but you also look if the abundances are the same. And then you would say like sample 1 and sample 2 here, they are similar because they contain the same taxa, but they're not exactly similar because the abundance is different. Like here we have 2 circles and here only 1 circle, and here we have 1 rectangle, and here we have 2. So that's something we can use. And basically, the most common ones you see usually are unifrac distances and Bray-Curtis. Bray-Curtis is really nice because you don't really have to build a tree or know what like taxa are similar or not. And unifrac actually incorporates that information. So unifrac actually kind of accounts for the fact that, yeah, there might be like different taxa in there, different bacteria, but maybe the bacteria that are in there are very similar. Like, for instance, there might be like different strains, or there might be like different species from the same genus. Like they're really, genetically, they're actually very like kind of replaceable. So you wouldn't, that would be like less impactful than, for instance, having like 2 completely different bacteria in 2 samples. And so how do you account for that? Well, it's done with a distance metric which is called unifrac. And just to give some intuition how that works. So again, let's assume 2 samples, and we often build up those phylogenetic trees. In those phylogenetic trees, basically on the leaves, we have the things that are most similar. So basically those 2 taxa here would be super similar, and then they group maybe with another set of 2 taxa that are also similar, and those taxa all together would also be kind of more similar than, for instance, those ones. So they kind of get separated by the tree. It starts like from a common node, or it doesn't even have to start from a common node. But the main idea is the lower you are in the tree, the closer you are to each other, the more similar you are, like phylogenetically. And if we talk about like phylogenetic similarity, one of the easiest things we can do is we can actually compare the actual sequence, right? So we have that variable region of the 16S gene, and often it's not exactly like that, but often the sequences are similar. That might mean that they're actually kind of are pretty close like in time. So they might not have mutated away like so long ago. So they're kind of more recently diverged than maybe ones that are really different. So we can kind of look at the sequences, and we can build those trees from like sequence alignments. So we can basically look, okay, those 2 have very similar variable regions, those 2 as well. So, and for unifrac, what we do is it kind of unifrac looks at that tree, and it kind of checks, okay, if I start from a single leaf, how long can I go up before I actually find like any branch that is colored like a different sample? So here, for instance, where they're really different, actually like all of the taxa that are similar in a single sample. So I can move up like the blue lines here. It's a pretty long path before I hit anything that will like intersect with like a red color here. Whereas like here we have two samples which are like phylogenetically very similar. So basically I move up here and now I already hit something that crosses with a red color. So that kind of means that in the lower branches, which means where things are actually more similar, we mix samples. So that means both of the samples contain more or less similar bacteria. Whereas in that case, both samples contain very different bacteria. Like all the bacteria separated quite big in that tree. So that's kind of what Unifrac does to kind of tell. And in the way that Unifrac, you also scale those, like the length of those paths by the abundances as well. So you make the paths like shorter or longer, depending on if you have a lot of the particular bacteria. Okay, that hopefully makes sense. If not, ask some questions in the Slack. So we did all that. So actually, if we run that diversity core metrics plugin, it actually generates a lot of different like distance metrics and analysis. So for instance, we do see that it did calculate the Shannon diversity index. It also calculated evenness, and it did also calculate some Unifrac distance matrices, and also generated some visualizations. So that might be quite interesting. Because that's like a good question. Like if we have those metrics, we actually want to look if there's differences in them, right? So that's something we can do. The first step we could do is, like we said, we could look at the alpha diversity, which is like the diversity within a sample. And we could, for instance, try to compare if it's different between the healthy individuals, the one with recurrent C. diff infections. So let's look at that a little bit. And let's run that statistical analysis. And that's done with the diversity plugin. And it has that alpha group significance, which actually will run like a statistical test. And like I said, we now actually have to, we want to incorporate metadata, right? We want to be able to tell what is a healthy and like a recurrent C. diff sample. So we also have to get in that metadata information, which contains that information basically. So if we run that, we actually get a visualization, which is called alpha groups, which will tell you to, which will help you to get that. So if we look here, that's in the diversity folder. You get that visualization alpha groups. You can download that again. You could again go to view.chime2.org, upload it here. And then now we actually see how that looks. And we can select that. So basically it gives you that box plot. So it shows you the Shannon entropy, which is the Shannon index. If it's higher, it means more diverse. And we see we have 4 with a recurrent C. diff infection, and they seem to actually have lower diversity. So that's kind of what we know about recurrent C. diff infection, honestly. We know that C. diff actually likes that setting where like not as many other bacteria are around. So it kind of is not really unexpected that we would see that. And it will also give you like a statistical test and a p-value. Here it does a Kruskal-Wallis, but it's only 2 groups. So that's actually the same as the Mann-Whitney test. So it would tell us, okay, or maybe we leave that on. So we do have some difference in alpha diversity, where individuals with recurrent C. diff infection have a lower alpha diversity. Now we might also want to look at beta diversity, but how do we visualize that? So we can start running that, and I will show you a slide and giving you some intuition. Yes, let's skip here. So for like principal coordinate analysis is like the standard way to visualize that. And what's the intuition is, you can see that as a type of an embedding. And an embedding usually means is, okay, if I have like all those differences between samples, could I arrange my sample, for instance, in like in a 2-dimensional space in a way that's consistent with those differences in beta diversity? So how would that work? Let's look at that example. So here I have like the beta diversity, right? So 0 would basically mean that's a distance matrix. So like the larger, the further away samples are. So obviously every sample is perfectly similar to itself, right? You are perfectly similar to yourself. So that's kind of intuitive. Then we see, for instance, that sample 1 and sample 2 are very different. Sample 3 and sample 1 are kind of close, and sample 2 and sample 3 are pretty close. And embedding, basically, the main idea is to kind of look at those and ask yourself, can I find an arrangement that's continuous That's like kind of consistent with that. So here we could kind of say, let's look at that arrangement, for instance. So here, sample 3 and sample 2 is really far away. That's kind of not consistent with that, right? Also, sample 2 and sample 1 should be really far away, and they aren't. So that's not really good. But we could look at the second setting, and it is fairly consistent. So here we see that sample 1 and sample 2 are pretty far away. Sample 1 and sample 3 are also somewhat a bit farther away, but a bit closer. And then like sample 2 and sample 3 are actually pretty close to each other. So that would be like better. And the basic idea of principal coordinate analysis is finding an arrangement that's like the most consistent with your initial distance matrix. And if we could look at that, it gives us a visualization, Permanova visualization in here, which is like the Permanova QZV in the diversity folder. Oh no, I just generated that in the main folder, sorry. So here we have that Permanova. If you run that code, we can again download it. Again, go back here, visualize it. Oh no, sorry, not the Permanova. My apologies. We want to actually look at the principal coordinate analysis, which are in the diversity folder, and you find quite a few of them, which is, for instance, we can look at the weighted unifrac. Oops, let's make that bigger. The visualization. So basically the results are just saved in an artifact, but then the visualization is always saved in a QZV file. And it's something called Emperor, and Emperor is just like a 3D visualization of that PCOA. So we can, for instance, download the weighted unifrac Emperor QZV. And if we do that, let's upload this one. To look at it. And we'll give you something like that. And you can actually go here and select what you want to color them by. So you could, for instance, color them by the disease status again, which will give you healthy in red, and blue is the recurrent C. diff infections. What we kind of see is there's definitely some separation going on. There's some interesting case here where one of the samples, which has recurrent C. diff, actually seems to cluster with the healthy individuals. Whereas like there's another group of 3 recurrent C. diff individuals are clearly separated here. So like 74% of that variation is like in that axis that separates them. And we can actually kind of look at it also in a statistical sense and run a statistical test on that. Basically, how does that work? So basically, that's a bit more complicated because often those things are not really normally distributed. So we, for instance, have seen the principal coordinate analysis. So kind of what you can test is, for instance, between the two groups, the healthy and the sick individuals, is there like a different centroids, or are they completely overlapping? And that you can then do with a method called Permanova, which is like a permuted analysis of variance. We do the permutation basically, since that like makes us a bit robust against assuming particular distributions. And the main idea is that you run like a normal multivariate regression model. So you would kind of say, okay, I have my beta diversity matrix, which basically tells us how different each of the single samples are. I kind of do for every column of that, I kind of have a regression model where I say, okay, are those differences actually explained by my particular metadata? So for instance, my disease status. So you could say that here, white is healthy. Well, maybe like white are controls, and then we have like recurrent C. diff infections. And that will give us somehow a quantity which is like a goodness of fit. Like it gets an F value, and like an F value is something like how much better is a model that incorporates that difference in disease status than a model that doesn't. But then like to actually compare to something, what I can do is I can just like randomly scramble those disease status. So like I kind of say, okay, now I randomly assign like the healthy status to some recurrent C. diff infections and so on. And I do that a few times, like a lot of times, and I get like F values from here, which are like from the wrong data. And I will kind of compare my real F value with that one. And that's something that's a bit more robust. And if we do that, like it's that code here where we run like the Adonis function. Adonis actually runs the Permanova. So we will have to tell us what distance metrics we want to use. So we're going to use the weighted unifrac distance again, because that's kind of incorporates a lot of information, incorporates information like which taxa are similar, and which ones are not. And it also takes into account the actual abundances because it's weighted. We again use our metadata file. We say we actually want to test on the disease status. That's kind of what we want to see. And it will generate that visualization. I ran it, but I can run it again. So if that gets run and we download that file, we can again take a look. And it will basically like give us a statistical test for the visualization, the principal coordinate analysis that we saw beforehand. So we actually opened that by accident before. So basically what it will tell us the F value. Like usually if that's bigger than 1, it means it's actually the model that includes that particular metadata is actually better. So that's what we usually want. You get an R squared value, and that tells you in that PCOA basically, or well, even in the real data, like the entire variance across the data, like how much of that is actually explained, for instance, by the disease status. In our case, about half of the variance in the entire data set is explained by the disease status. And you get a p-value again, which is kind of like, you can say like, it gives you like confidence that this is actually like a result that is like, yeah, that is there. And that is, in this case, it's okay. So it kind of tells you, okay, we do see that even in better diversity, we see like a significant separation between those samples, between healthy and sick individuals. We actually saw there was like one sick individual that seems to be closer to the healthy ones, but on a global level, they're pretty separated. So they do have different compositions. So, okay, we know now that they're different, they have different alpha diversity and beta diversity. So the next thing we might want to look at is the taxonomy. That's what we did. And that is basically related to the question, what organisms are actually there in our sample? So now where we know they are different, we actually want to know where those differences are, what are the organisms? So how would you go from a sequence to an organism's name? Like how would we actually be able to tell that, for instance, a Clostridium read? And basically what we want to do is we want to take our amplicon sequence variants and want to assign like a taxonomy to it. And the taxonomy is kind of like a hierarchical system that classifies basically in organisms. For instance, yeah, if we have, we usually do that like, for instance, on the kingdom. So we usually for bacteria, the kingdom would be bacterium. We have like a phylum, class, order, and like it gets kind of more fine-grained the lower we go to kind of get down there. And that's kind of what we actually want to get. So how do we assign that taxonomy to our amplicon sequence variant? And the most obvious thing is like, we could say, if we already know the sequence from a known organisms, why not just compare to that one? And that's something we could do. In practice, it doesn't really work that well. And why doesn't it work well? Well, because those like model organisms are often not really that similar to what you have in your gut, for instance. And that can actually create some bias in there. Also, you might actually be not as powerful there because you might have sequenced only one species from a particular genus, which is like not as representative, also just doesn't generalize that well. So actually you can get better by doing like a different approach. And that is called like a multinomial naive Bayes. It's a way to classify sequences into taxonomy with a bit more robustness. And the basic idea is that we work on something which is called K-mers. And K-mers are just subsequences. For instance, a 3-mer are all possible sequences that are only 3 nucleotides long. And we can actually enlist them. So if we have like a query sequence and I kind of want to know what taxon that is, I can actually generate all the K-mers from there, like ACG, CCG, GCG, and so on. And I can count them. And then I can go through my database and I can also do the same thing for the known taxonomy, right? Let's see for taxon 1, we actually have a distribution where we know, okay, the ACG subsequence appears like in 25% of the cases. And then the CCG1 in 25% as well. and the last one in 50% of the cases. Also, that taxon in my database is there in like 20% of the cases, which that's often called a prior. That's like the the prior probability of just finding that taxon in your reference database. And I do that for both taxa, and then basically I can kind of say, okay, what's the probability that if that sequence actually came from taxon 1, what's the probability of generating that particular subsequence distribution? And you just go through it. So you start with a primer and then you multiply with what you actually observed in there. So for instance, we observed the ACG sequence once, so we multiply once with 0.25. We observed the CGC sequence twice, so we multiplied 2 times with it, which is basically just squaring the 0.25. And we observed CGC once, so we also multiply with 0.5. And that gives us like the over probability to kind of observe that, that query under the taxon. And here the trick is actually in base. So the base basically tells us that you can turn that around. So if you actually only have the probability of a query assuming a certain taxon, there's actually a way to turn that around into the probability of observing a particular taxon if you observe a certain query, which is what we actually have, right? The query is what we saw, that's the amplicon sequence variance, and we kind of want to get the taxon. So we want to have what's on the left side here, but what we actually can get is only the part here on the right side. We can actually ignore the probability of the query part here because that won't really matter. Like it would still be comparable between different taxa. So if you do that, you can basically calculate it for both taxa, and then you can compare. And you basically see that the first taxon is actually more likely. So you choose that one. You would say that query comes from the first taxon. And that provides better generalization, since it kind of takes like a more overarching principle of saying, okay, I assume that bacteria will use kind of the similar k-mers. That's actually gives you much better classification, and we can run that as well. Obviously, what we will need for that is we kind of need those reference database. I see that we're like already at 11. We'll probably go a few minutes over to finish that course. And since the exercise is actually really short, so that will probably work out okay. So we will need the reference database first. So we will start by downloading it. That kind of has those reference k-mer counts, basically. It is like a large database, has a lot of different bacteria in there. And it's actually uses the same primers, so it's kind of cool. They use the same variable region and they counted like all the k-mers and made those reference distribution for us with like the entire taxonomic classification. So that's often taken from sequence organisms that were already known. And then we can use that reference database and use the classify function in QIIME 2 from the feature classifier plugin. So where we're actually giving it now the amplicon sequence variants that we got from Data 2. The prior is identified by basically looking at the database and just checking like how many sequences from that taxon you have in that database. So for instance, if the green genes database, which is the one we downloaded, has like 50% would be Bacteroides, that would be the prior. And then we kind of use that classifier with the naive Bayes classifier, which is like a pre-trained classifier that we downloaded, and we get like as our output the actual taxa distribution. So now we know what each the taxonomy of each amplicon sequence variant. So if we run that, this will not take too long. There's larger databases you can use. Green genes by this point is a bit outdated, and it doesn't contain as many reference data. So you could also use the silver database. That's also downloadable actually by the same link I put in here. If you go through it again, it has that link of the data resources, but you can also find the silver database. That's an updated database which has way more sequences in there. So that's kind of predicting it here. And we could have like a look as soon as that finishes, which shouldn't take extremely long. Okay, so now we actually have the taxonomy. So actually, let's look at that. And we will do a bar plot. Basically, that will tell us like what's kind of in each sample and different taxonomy levels. So we generate the bar plot. We can look at that. It's called taxa bar plot. Yet again, we can download it and look at it. Okay, let's open that up. Okay, so first thing, it will show basically here each single sample. And we start out actually with level 1. And that in this case actually pertains to the kingdom. So we sequence basically bacteria. So it's not really surprising to see that in each sample, 100% of the reads came from bacteria. We can actually choose a lower level. So one that is often used is the phylum level. And that already gives us an idea what we have in here. So for instance, usually we see that the major phyla and basically all samples are Bacteroidetes and Firmicutes. Those are like the major players in our gut microbiome. And then we also have some proteobacteria. Proteobacteria can be good for you, but some are bad. For instance, Clostridium, E. coli falls in that class as well, and some other ones usually are not super healthy. We also see some fusobacteria, and the one thing we already see is that there's definitely something going on. Like those distributions, they definitely do not look the same across samples. So we have a few here where we have pretty high proteobacteria ratios, whereas in others we have more Bacteroidetes and actually lower proteobacteria levels. And you can actually choose a color palette that actually, oops, sorry, you can actually use the metadata in here. So for instance, we could also sort the data by the disease status. So now we kind of see it starts with the 4 healthy ones, and then there comes the recurrent C. diff infection ones. And we definitely see, okay, so it looks like for most of the healthy ones, we kind of have a lot of Bacteroidetes and Firmicutes, and then we see some actinobacteria. But for like the individuals with recurrent C. diff infection, we actually see that kind of a big Microbia abundances in here, and then sometimes a lot of fusobacteria. And fusobacterium, especially genus, is usually bad news. So fusobacteria are pretty abundant in our mouths. They're usually not in our gut by those large abundances. And a lot of fusobacteria in our gut are really bad because they're really related to colorectal cancer. So colorectal cancer can usually be, you can actually see big expansion of fusobacterium. So that usually means if you would see something like that, like the last individual with a recurrent C. diff infection here, that doesn't look too good. Like it's a microbiome dominated by proteobacteria and also by fusobacteria. And those are two things you don't want to have in those high abundances. Okay, we did that. And we're pretty much through with that part here. I'm just going to show you like a small part how you can actually look at that data also directly using it from QIIME2. So for instance, we saw those things. And the bar plot is nice, but we might visualize it a little bit different, right? So for instance, we might be interested to go lower in the level and actually look at genera abundances. But if we look at that in a bar plot, since there's so many different genera and they like have differences like maybe 3 orders of magnitude, it might be hard to visualize them. So let's first get the actual genus level data and we can collapse it. So basically we take all the data and we count how often each genus appears in every single sample. And that can be done with the QIIME taxa collapse comment. So now that data is a QIIME artifact, but this time we actually want to use it ourselves. So we want to analyze it ourselves, for instance. So we kind of want to get to that data into a format that we can actually understand and read. So the first thing we can do is we can export it. And export basically just means unzipping that data file and giving you the data that's actually contained in there. And then we will actually use a comment, which is like the biome convert comment, since that table, that abundance table on the genus level is like in a format that calls biome, which is like a really efficient format to even save like large abundance tables. And we actually generated and converted to a tab separated file on the genus level. So that's something we can definitely parse and read with a lot of different libraries. So that's kind of what we could do as well. There's always a pass from like a QIIME artifact to get the actual raw data. So we get the raw data out of the QIIME artifact into a TSV formatted file. And now we could, for instance, use Python. And like, there's not a necessity to understand that 100%. If you're familiar with like doing visualization with Python, that might be pretty straightforward. But basically just to show you that's something you can use with R, with Python, with whatever you like to analyze data. So here, for instance, we can read the abundances now with pandas. So we have like a normal tab separated file. We will skip the first row because there's like a comment in there. That TSV file actually has like the the entire taxonomy down to the genus level. And it's like separated by semicolons, so we can strip that apart and get like a full level of taxonomy. We actually get the one that pertains to the genus name. We also like kind of remove certain parts of it. Like that's basically just cleans up the data a little bit. That's what you would often do. And we, for instance, we can do whatever transformation we want on that raw data. So for instance, we said, okay, they're different by several orders of magnitude. So maybe let's like log transform them. But also we know that the total number of reads in each sample is different, right? So we had a sample with like almost 100,000 reads, one with like 10,000 reads. So that's the thing, like for instance, if I observe that Clostridium has an abundance of 2,000 reads and a 10,000 read sample, that's like 20%, right? So if it would have 20,000 reads and the 100,000 read sample, that would also be 20%. But if we just compare the raw values, 2,000 against 20,000, we might think there's way more. There isn't actually, just that we like by some artifact in the sequencing got more reads. So we can kind of like subtract also the logarithm of the like the mean abundance and kind of even out that effect of the library size. Library size means just how many reads you had in each sample. Usually it's not really meaningful, like it doesn't mean anything, just an artifact of the sequencing process. And that kind of particular trick here, we add 0.5 because abundances could be 0. You can take a logarithm of something that's 0. Basically, that transformation here is called the center log ratio transform. It's pretty popular for microbiome data because it has some good properties, and it kind of really makes it a bit pop more visually. And if we have that, for instance, we could use any visualization library like Seaborn to actually generate like a nice heat map of the data. So in our case, it would look like that now. So we get here every single genus in our data set. That's quite a few of them. And we kind of see them by each sample, and it even clustered them together. So basically, like individuals with similar microbial profiles are kind of close to each other in the rows, and microbes that co-occur in like the same individuals are also close in the columns. So it kind of gives us like a good step to kind of see those patterns here. For instance, we see, and that's actually something we already saw before, we have only 3 of those recurrent C. diff individuals actually kind of look similar, and then one of them is actually very similar, very different, and that's the 48 one here. And we do see that it appears that between them there's this entire set of bacterial genera that seems to be really lower abundance in the individuals with recurrent C. diff infections. And there's a few of them that seem to be higher, which are the ones here. So we give Haemophilus, Veillonella, for instance, Proteus, that's all higher abundant, like more yellow means more abundant. And that kind of gives us like an idea that those might be more abundant recurrent C. diff individuals. And like, yeah, that particular intermediate case here is quite interesting because this one is an individual with recurrent C. diff infection. It does seem to have a lot of the kind of bacteria that associate with the healthy individual, like we see like Roseburia and Bacteroides, for instance, which are usually good for you. But it also seems to have quite a bit higher abundance of the ones that are actually specific for the recurrent C. diff infection. So that's like an interesting case, for instance. We will look into that particular individual a little bit in the next course part. Okay, yeah, sorry for going a little bit over, but we're through it here. It might have been a bit quick in the second half, so feel free to ask some questions and we can go through it. We actually have quite some time now, so we will go through, I think, 11:45, if I'm correct, with the exercise part. The exercises are fairly short, and also the expectation is that you only do one of the 2 exercises. I put 2 here, so you have something you can do at home. And basically, you can either do exercise 1, which is like sample classification. And that's like the question is, okay, can we actually kind of predict a person's recurrent C. diff state just by looking at the microbiome data? And we will use the metadata file. And here I just showed you how that looks like. For instance, that was the metadata file that we had before. So it gives just each sample ID and the disease status and also some descriptions in here. Usually the healthy individuals are actually donors for fecal microbial transplant. So they are not only healthy, they are kind of healthy and have super healthy poop. They have super poops. And then like you can use a Chime plugin called Classify Samples. So if you want to actually understand like what plugins they are, like I said in the beginning of it, I actually gave you some links to documentation. So you could, for instance, go to the Chime2 website, chime2.org, go onto the documentation, and then you have that part plugins, which shows you everything that Chime2 can basically do. So I said, okay, we want to classify. So we could look for, where do we have it? It's sample classifier plugin, for instance, and will actually tell you, okay, what can you do with that? And what particular pipelines do we have? For instance, if you go on classify sample, it will give you a nice introduction of the entire comment, how to call it, what are the meanings of the individual arguments. You should have everything you need. So the feature table is what we generated in Data 2. So you could classify those samples here. Or alternatively, if you kind of want to look into like the phylogeny a little bit more, you can use the ITOL website here and upload like the tree that we built. We actually haven't looked at that tree so much, like what is the relation between different variable regions that we found. And you can annotate it, for instance, with the taxonomy, not the abundances. So both of that can be used. So you can pick either one of those and do it. It should actually not take too long. So we should be all set for 11:45, and then I will show you some solutions here and how that might look. Okay, thanks so much. So we will be doing that particular break. Obviously, you can also use it to maybe grab a coffee or go to the bathroom, generate some super poop yourselves. And then you can get to one of the exercises, do that, and we will be back at 11:45 and looking at the exercise solutions a little bit. Thanks so much for bearing with me. Sorry for going a little bit fast in the second part. Hopefully we'll do better in the next part of the course, but yeah, I think it was fun, at least was fun for me, and thanks so much for joining us. See you later. Okay, everybody. So I hope you all had some time to look at the exercises. And I will just quickly show you some example solutions, maybe discuss like some intricacies here. So classify samples. After we classify samples, the first like exercise was to basically classify the samples using the data. And maybe the first traps that I put here is I didn't really tell you what table to use. So there were quite some strategies here. You could have used the abundance table that you got from data 2. You could have used the genus level table that we had, or you could have used the rarefied table from the diversity analysis. And rarefied basically means that you subsampled each of the samples in a way that they have exactly the same number of reads. Which can have advantages and disadvantages. The obvious advantage is that now every single sample contains exactly the same total number of reads, so you kind of remove the effect of library size. But also if you have, for instance, verify everything to 10,000 reads and you had 100,000 reads in a sample, I mean, you're kind of only analyzing 10,000 of the 100,000 reads in there. So you lose some data there. This is why like there's always like kind of an, like a trade-off between kind of using rarefied data or maybe log transforming it and all of that. In this case, like if you did it with the rarefied table, that's fine. You could also use the genus table. And then you would have put in the metadata file again. And that metadata file in the beginning we saw had that column disease status that told us who's healthy and who is actually has a recurrent C. diff infection. And that's kind of what we wanted to predict, right? So that's what you would have put as metadata column. That's the output. And I also put like an n jobs parameter here of 2. That's kind of how many CPUs you want to use to run that analysis. It can make it a bit faster. In the Google Colab environment, we have 2 CPUs. I mentioned that in the beginning really shortly. So I'm going to use 2 CPUs here. Sorry. And then we have our output directory, which is basically the classifier directory where we would have saved the data, for instance. And I think most of you actually managed to run something very similar. And it would have given you quite some files you could look at. I told you to look at the heat map visualization, which kind of looked similar to what we generated beforehand, which was kind of that thing here. But now either it would have done that for each individual amplicon sequence variant. If you would have used the genus level data, it might have looked very similar, but you only probably use 2 rows, sorry, because it kind of summarizes that across all samples, like it's kind of either 2 example samples or the mean. And then you could have also looked at the accuracy, and that would have told you like how well that actually does in predicting the C. diff state. That would have been the accuracy results visualizations. It actually would have told you that's almost perfect in differentiating sick from healthy individuals. But there's a little problem here, and that's that we only have 8 samples in our data set. So that's not really a big study. Like most 16S amplicon sequencing studies those days have almost thousands of samples, hundreds to thousands. That's like one of the largest American gut project by now has up to 30,000 samples, for instance. So that's like a very different dimension of data points that we have here. And it turns out that we often get that problem with like low number of samples, we get like overfitting. So basically, since we kind of, the machine learning methods that kind of does that classification has a lot of different parameters it can fit to the data, way more than we actually have data points. So it kind of over captures the actual data. It doesn't generalize well. So if you would give it like data that's not from that experiment, it would probably do a pretty bad job of classifying it. And we will look at that a little bit in the second part of the course, like because we do something similar with the Microbiome analysis. And I will show you that there is a difference between using the entire data, training data, or kind of doing it with data that hasn't been used for training. So we would do a decent job. We kind of saw that. We probably would still have a problem with correctly classifying that one recurrent C. diff individual that looked almost like a normal, healthy person. And in the second exercise, if you wanted to do that, I kind of had you like visualize the phylogenetic tree a little bit. And that's kind of what you can get in the final output. What I did here is basically I loaded the tree in there, and then I actually said ignore the branch lengths. So yours might look like a distorted version of it. What we have here is often called a cladogram. So it's still exactly the same thing. If 2 things are close, they are still very similar. But the length of the branch is not directly associated for sequence to sequence similarity again. It's just like a way to kind of show it so it all has the same maximum length basically here. And then the first thing that happened is if you imported the taxonomy data, it actually changed the names of all the leaves, where before you actually had those like weird random IDs for the amplicon sequence variants, and now got substituted with the actual taxonomic classification. So if you zoom in that on ITOL, you can actually see exactly what's the classification for each of the individual amplicon sequence variants. And one thing that was pretty obvious here is if you look at a few of them, you get like that they're only classified at a very high level. Like you get no genus or not even a family for them. And that happens as well. If there's like no sequence in the database, that's pretty informative for that amplicon sequence variant. It might just tell you, oh, well, it's a bacteria, but I don't know more than that about it. So that was also to kind of see that you will get like different levels of taxonomy for each of them. And then if you actually imported, for instance, the rarefied abundances, you kind of saw that each of them was kind of annotated with the abundance in each sample. So you could have seen, for instance, that there's one ASV that's actually kind of dominating almost. It's like really abundant. I think that's a Bacteroides, I think. And you also saw that there's, for instance, others that might only be present in a single sample, for instance, and not really in any others. And why did we use the rarefied table? Well, again, if we wouldn't have corrected for the library size, so the total number of reads in each sample, this might be misrepresented a little bit. For instance, if I see like more red here, but that comes from a sample that had like 10 times more reads than any other sample, it might actually mean that like by proportion, that's actually not that abundant in the sample, but still just because we had more reads in that sample, it gets overrepresented here. There's other ways to do that. You could have, like, for instance, you can do relative abundances where you kind of divide by the overall number of reads. You can do the centered log ratio transform that we did before, but all of that would have been harder to get into ITOL, which is why I recommended to use the rarefied tables here. Okay, yeah, that was it for the exercises here. If you have additional questions, you can still put them in the Slack and we will respond to them. But actually, just to motivate the second session a little bit. So we're actually at a pretty good stage right now. So we started out with like weird raw sequencing data and no human would have made any sense out of that. And by now we have a pretty good understanding what's going on. Like for instance, we have seen that individuals with recurrent C. diff infection have a lower diversity. So there's less, there's kind of, like they're less diverse in the individual samples. We also saw that they really are different from healthy individuals, like in better diversity. So there is like in a global microbiome states, they're kind of very different from each other, except for one sick individual that actually looked more like it was a healthy individual. And then if we looked at the genera, we did see that there are like clusters of genera that like seem to be kind of more associated with the disease and others that are more associated with the healthy state. So the obvious question now is kind of how does that work, right? Like, are those differences in taxa abundances here, do they actually have a metabolic consequence? That's really what we might be interested in. Because, yeah, it's cool to see that there are bacteria there, but in the end, we as scientists or we just curious and we want to understand what's really going on. So how does that kind of impact that C. diff can actually get into that system and infect? Because, I mean, you might say, oh, okay, if you have Bacteroides, it seems to be protective, but how does it do that, right? So we might want to be really interested in, is there something that Bacteroides does? Does it produce a metabolite that makes it hard for C. diff, or what's really going on in that system? And that's kind of a question of functional analysis. So now you want to see what is the function perpetrated by the microbiome that actually enables, for instance, a healthy state or makes you less healthy. And that's what we'll be looking at in the second point. So there's actually no code for the cladogram. It's just an option. So if you open that tree in ITOL, maybe I can show that here a little bit. So basically what you probably did is you download either the rooted tree or the tree. Honestly, it makes no difference for that particular step. You go to the ITOL website. And you probably uploaded it here. You could have put in a name if you wanted to, but you just go and upload. You choose choose the file, sorry, that tree file, for instance. You uploaded it, and then you kind of saw your tree. And sometimes it's like kind of hard to see what's going on in those short regions here. So that's now the length is actually scaled by the actual difference in sequences. So you could kind of see that those things are fairly similar, but there's like that section of the tree that's really different. And if you kind of want to see that without the branch length, you just in that branch length option here, you say ignore. And it will kind of give you that cladogram where it's kind of rescaled a little bit. And then like what you probably did is take that plus button, upload the other data to kind of visualize it. You can also make it like non-circular, which sometimes is nicer if you want to read the labels. Then you can zoom in with your mouse wheel, for instance, which gives you like the same tree, but it's kind of aligned in a non-circular fashion. And yeah, we can basically, yeah, we can definitely post the code for the classifier. Some of the TAs can probably do that for whatever they ran. Yeah, we can also provide the solutions to you. Cool, yeah, thanks so much. So now we are kind of finished with that section. Awesome participation so far. I'm really happy with how everyone is asking questions on Slack. Like it's super cool to see so much curiosity and like also like very intelligent questions there. So we are actually having a lunch break now, which might not be lunch for you. So I'm sorry if you're in a different time zone, it might actually be inconvenient, but we'll have like a half an hour break right now. And then we'll be back at 12:30 our time or in half an hour from now, basically to start with the Microbiome tutorial. Thanks so much. And yeah, glad to have you all here. Okay, everybody, welcome back to our second session, which will be named Modeling Microbiota Wide Metabolism with MyCom. And as we motivated it, we were now going to take a little look into like the functional consequences of the different microbiota we observed in the first half of the course. Also, if you joining just for that second part of the course, that just reiterates that we are also aware that like a lot of people might be in different time zones right now. So all of those course materials, including like the presentation I'm giving right now, will be available as videos. The presentations are available online, the notebooks are available online, and the Slack, the support channel, will also remain open for, I think, at least this week. So if you're, for instance, like if it's 2 o'clock in the morning, like for you right now, and you're basically dying of tiredness, like you can also just continue doing that course tomorrow, and you will just receive the same support. You can still do the exercises and ask for help. Will all work out. So don't worry about it. Okay, well, let's get started with that second part of the course. And again, like before, like it might be helpful to actually open up the slides, and I'll ask the TAs maybe to just post that link again. Actually, if you have the previous slides open, it's actually very simple. You can go to the address bar and change the last part, which was 16S, to MyCom now, and it will just open up those slides for you. If you don't, you can just click on the link in the Slack or the one you received in the email or on the course website. It's on all of those. And just a quick reminder, and maybe also for people that have only joined for the second part, we have those 3 parts that will be running. So there will be that presentation that I'll be using to explain things, but we will be actually running the analysis in our Google Colab notebook, which we will open in a second. And we also have that chat. So if you haven't joined the support Slack, please do so. You'll find that link in the chat of that Zoom call, and also in this in the email that was sent to you, and that you can ask any questions there. Those will be relayed to me if something like really bad happens, but you also get like immediate support from our TAs with anything you might find a bit difficult to follow. Okay, so now we will be opening again like a notebook, but now we'll open a notebook for the second part of the course. I'll be using that link here, but you can also use the link that is probably posted in the Slack right now, or the one you received in the email. So, and we will just doing exactly the same thing that we did before. So we're opening up that notebook. It looks pretty similar. Some more explainers here. And again, like the structure is the same. We have those cells that are either text and we have parts that are code. In this step, for most of the course, we'll actually be running Python code. So we won't need that initial exclamation mark, and only for the exclamation mark, only for the first part where we install some things, and later on we actually use pure Python code. And that thing works the same. If you want to connect to the runtime, click the connect button on the upper right here. If you don't see the connect button, it shows you something like RAM and disk. You're already connected, so don't worry about it. And if you're connected, it should look like that. So you see the free RAM and the disk space you have. And again, on the left side, we kind of have the table of contents. On the lower region, we have that folder symbol that shows us all the files in the notebook, which we'll be using. It's the same thing. But yeah, we'll be using it in a second. Right now, let's start with a little bit of introduction, what we actually want to do right now. So I already said that in that second part, we were really interested in function analysis. So we analyzed our microbiome data. So we basically went from raw sequencing data to abundances of microbes. We know which microbes they are. We have the taxonomy. But taxonomy isn't really a perfect explanation of what's really happening in the system, right? So we might be interested, okay, like if there are like particular microbes associated with a disease, why is that so? Like what is the actual mechanism that underlies that? Is there, for instance, a metabolic interaction, which is what we mostly will look at? And there's several approaches to that, which I would say there's basically the most common ones that we've used a lot is basically gene abundances. For that, you basically need to do metagenomics. Everything else doesn't really work that well in practice. Well, somewhat. So for metagenomics, you, like we said in the first course, you actually try to sequence entire genomes of everything that's in our sample. And if you do that, you can simply count the genes that are in there. There's certain analysis pipelines that basically go through that. For bacteria, it's not really hard to find out what a gene is because they always start kind of with a similar type of promoter. So it's really easy to identify open reading frames. So you can do that de novo. You don't even have to know what that gene does. Then often you compare those genes that you found to databases and you can quantify, okay, that particular phosphofructokinase is pretty abundant in that sample, for instance. That gives you genes and gene abundances, and that can be pretty informative, but it has like a small caveat, and that is that gene content is basically only a capacity or potential. So it tells you that the gene is there. But for instance, you might have a gene that helps you to break down a particular starch, but if you don't eat starches, that gene just sits there, right? It's not going to do anything in your gut because it's never really encountered any starch, so there's no effect to it. And that's actually quite common in bacteria. Bacteria are really good in adapting to different environments, so they kind of have a lot of genes to deal with different environments. But if they don't find that environment, those genes will just sit there. They might not actually do something. So that's something we have to keep in mind. Other more elaborate methods, you can use metatranscriptomics. Then you see that genes are actually expressed, which is already a bit better than just having the gene, because the gene might be there, but it might be in an operon that's switched off. So you're actually not even expressing the gene at all. There's no associated protein. You could use proteomics and look at the proteins that are there, which is like even better to see if there's certain enzymes that are actually present, and even do metabolomics where you actually measure metabolites. And if you measure metabolites, you could kind of see if there's particular metabolites that associate with certain microbes, for instance. All of that has a few caveats, and I will kind of start out with a bit of a hot take here. And sorry if anyone feels like offended. It's not really meant really mean, but like just saying that genes and metabolite abundance are really cool, but they might not be what you really care about. And why is that? Well, for instance, I could go home and eat a bit of kimchi, which has like a lot of really good bacteria. And those are bacteria you might not actually find in our gut that commonly if you don't eat kimchi. Then afterwards, I eat the bubble gum, and because I'm not really careful, I swallow it. So now you will see that certain kimchi bacteria and the bubble gum are both present in my gut, maybe in high abundances. I would even say they're related to each other. And we conclude from that the kimchi bacteria are producing bubblegum in my gut, which would be a quite interesting paper, but probably not that one that would really be considered like good science. So just because two things co-occur doesn't mean that one, for instance, drives the other, right? So how do we do that? Like, what is the quantity we actually want to see? And for that, we have to look a little bit how metabolism usually works. So what we might be way more interested in are fluxes. So fluxes is actually a transport of mass across a system. So a flux is a rate of mass conversion, whereas concentration from metabolites are usually measured, for instance, in millimolar. Flux is actually measured in millimolar per time unit, for instance, millimolar per hour. And for bacteria, we usually also normalize by the gram dry weight of the bacteria that is present in the system. Why is that? Well, because 10 bacteria probably eat way less metabolites than 1 million bacteria, right? So we kind of have to normalize that a little bit. Fluxes are like super informative because as we see on the right, that's actually what's happening in the system. We can actually see the flow of mass through the entire system. So we can, for instance, see that like a lot of glucose gets converted to pyruvate in that example. And that's kind of one of the caveats if you measure metabolite concentrations, for instance, since glucose gets, for instance, absorbed by bacteria so quickly, they love to eat glucose. They're really like sugar freaks. So you actually don't see high concentrations of glucose in your gut usually. Whenever it comes in there, it's either your gut will take it up or the bacteria will eat it up, but the concentrations will be really low. So, and that's maybe a bit counterintuitive, right? It's like one of the most important metabolites for that system, but it's really not there, like, or measurable in that abundance. So that's kind of what we have to take into account. But if we would look at a flux, we would see that there's a massive transport of glucose into bacteria. So that would really tell us, okay, that's something that's important. So fluxes are super interesting, and honestly, that's like not new. We have known that for quite some decades that fluxes are cool, but the problem is that they're really kind of expensive to measure. So one thing I could do, I could longitudinal metabolomics. So if we go back to our bubblegum example, what I could do is I could eat kimchi And then I could kind of track myself, like every hour I kind of measure all the metabolites in my gut and I see if there's actually bubblegum produced. Like I start off with little bubblegum and over time it gets more. Then I could kind of say, oh, those bacteria really produce it. That is actually kind of a flux, right? Because I could say, oh, I produce like, I don't know, 2 bars of bubblegum every hour in my gut. So that would be like more of an evidence that it's really produced. There's also stuff, also methods, not stuff, that could produce fluxes, and you don't even have to measure like longitudinally. And that's basically the trick is you can give, for instance, glucose to bacteria, and then you label the carbon like with your radioactive probe. You make it radioactive, so you can actually probe it. And then if you look after a while, you take a single sample and you can actually just check, okay, I know I gave a glucose where every carbon was kind of radioactive, right? So now I look at pyruvate and see of all the pyruvate in the system, like how much of that pyruvate actually has like a radioactive carbon. And that will kind of tell us like how much was converted to pyruvate. So that's like another trick. And that's even more expensive, unfortunately, than longitudinal metabolomics. So there's definitely a cost factor. 16S sequencing, for instance, can be done really cheaply, but that is really hard. And that was kind of the stage which we started as well. We kind of wanted to get like functional insights, but often what we had was like, well, in good cases, metagenomics, in bad cases, only 16S data. And like I said, for gene content, you actually do need metagenomics. So that functional analysis wasn't possible. There's some tools like PyCrust, which kind of like, what they do is if you know that there's a certain taxon and you have a reference genome, you can kind of predict what the gene content might be, but the taxa in your gut might actually not be exactly the same as the ones that were sequenced, right? Like mostly in the lab, we maintain like lab strains, which are like really easy to grow in the lab, but they might not actually be super informative. So that's something we have to account for here. But yeah, we're still interested in flux, as we said, right? Because we might only have 16S data or metagenomics. So how do we get there? And that's basically what flux balance analysis does. Flux balance analysis is actually a computational method. And it's an input. It can go from actually genomic data. And the question is, can we infer the most likely fluxes in a biological system? And it has been used quite extensively, but we haven't seen it that much in microbiome science, but it has been used a lot in bioengineering. Like a lot of chemicals those days are produced by bacteria that are kept in really huge tanks. And to kind of design a bacteria that produces a lot of something, you want to maximize the flux, right? So that's where they use flux balance analysis to actually like find particular mutations that they can introduce in the bacteria so it produces the actual chemical that you want. So how does it actually work? Like how can we computationally get like fluxes? And there's a certain assumptions that we make and like certain concepts that we will introduce. So the first one is kind of the flux cone. So the first thing that we often assume is that we have like a constant flow, or what is often called a steady state. And that can be imagined a little bit by if you open your windows right now to like let air into your room, which is especially during a pandemic, a pretty good idea, right? So if you open that window, air will come into your room, but it also obviously air will come out, right? You exchange like old air with fresh air, basically. And while that happens, obviously there's a kind of a balance, because if air would just be coming in and not the same amount of air is getting out, well, the apartment would blow up, right? It would just like fill up with air like a balloon, and at some point it would pop. It could also be the opposite. Air is coming in and more air than is coming in is actually getting out, which would create a vacuum, and then it would implode. So both of those obviously don't happen. Like nobody had his apartment blow up just by opening a window, at least if nothing is in front of the window. And so there is a certain balance that stuff that's coming in is also kind of equilibrated by stuff that's going out. And that actually works for a lot of physical systems, and it kind of also works like that for metabolism. For instance, like in our state, like bacteria grow in our gut, but then we also poop. So it's kind of like an equilibrium with those two processes. And an example, we can actually formalize that. So for instance, we could look at that example biochemical reaction or chemical reaction here, where we have 2 hydrogen molecules, like with an oxygen molecule, and kind of converts to 2 water molecules. And if those fluxes are imbalanced, like what I can do is I can basically, everything that's produced in the reaction, I give it like a positive sign and multiply it by the stoichiometry of the reaction. Everything else I give a negative sign because it gets consumed, so we have less of it. And I would say that they're balanced. So the entire balance of it must be 0. So that's what we see in the balance equations. And then we can also put constraints on it. For instance, we can say, okay, in this reaction, I actually want water to be produced. And the reaction should only go in one single direction. So it can only go from hydrogen and oxygen to water, but water cannot like deassemble into oxygen and hydrogen. So, and then that would basically mean I say, okay, that particular flux is larger than 0, for instance. That's like maybe not something that happens in bacteria, that particular reaction, mostly because of the huge explosion that it entails. But that particular chemical reaction is a reason that why we use helium in balloons and air chips and not hydrogen anymore. So what that actually means is that there's like a limitation, since like I need a particular balance of hydrogen and oxygen to kind of form like water. It's not all possible values of them are possible, right? Like if I have like 1 hydrogen and 1 oxygen atom, can't really produce 10 water molecules. So that's kind of a limitation, and that can be formalized like by like a set of linear equations that kind of limit the space of possible fluxes. Like there's only few flux distributions that are actually valid in that setting. And I could put constraints on it, like for individual fluxes, I could say like they can also go in a reverse reaction, then they will become negative. Usually we split up those reversible biochemical reactions, say that's a forward and reverse direction. And so that's one reaction for each. The flux are usually positive. And if I do that, then basically one trivial solution of that equation is that all fluxes are 0. If you multiply anything with 0, obviously it's 0. So the 0 point is definitely included. And then it feathers out into that flux cone, which is the space of all possible fluxes. And flux balance analysis is basically hopefully trying to find that one single point in that space that's actually the most biologically relevant one. So how do we make it more relevant? Well, we can actually look at growth, right? Because if we know, for instance, bacteria present in our system, well, they can only be present if they can grow. Otherwise, they would be pooped out, for instance, by us, and then they would be gone at some point. So one assumption we can basically make is that we can say, okay, we want to find basically that part of the flux cone that corresponds to production of maximum biomass. And that's fairly easy. Biomass is essentially a flux. It requires a lot of components that have to be coming in to continue to produce biomass and let the bacteria multiply. And if that actually gives like a subspace. So in that flux cone, we have like that red area that corresponds to maximum biomass. And the cool thing about flux balance analysis is that we can incorporate a lot of information. Like we incorporate all the biochemical stoichiometry, and that we can actually get just from a genome. So if you have a reference genome from organism, we can look at all the enzymes. For most of the enzymes, you kind of know which biochemical reactions they catalyze and what's the stoichiometry of that reaction. And by that, we can infer like that entire stoichiometric matrix. And like I said, we make that steady state assumption. And in practice, for bacterial growth, that kind of means we're kind of in the exponential phase, because we're in a phase where the growth rate is constant. If we produce biomass as a constant rate, our growth rate is constant. So we're kind of in that like exponential growth phase of bacteria. And bacteria like to spend a lot of time in that growth phase if they can, but it's an assumption we have to make. And reversibility for reaction, that can usually be determined by thermodynamics. We know that certain reaction can only happen in one direction, others don't. But I also have like fluxes that actually symbolize like import in the system, so things that are coming into the system from the outside. So I can actually incorporate the environment as well. So for instance, I could model that with different diets. So that's like a little bit something that would be really hard with metagenomic data, because you only have gene content. You don't really like think about how the diet might influence that system. So we have that sub area that's maximum biomass, and now like a lot of flux balance analysis about like an art of finding either a range or like even a single point in that space that's like the most representative one. One that actually works really well is finding a parsimonious solution. Parsimonious kind of means find one that's kind of uses the least amount of enzyme. And it basically means I try to keep most of the fluxes as small as possible. And why is that? Because the flux itself is a function of the substrate that's available, how much substrate do you have, but also how much enzyme do you have. And obviously it's costly for bacteria to produce more enzyme than they actually need. So if they can get maximum growth by producing the minimum amount of enzyme, that's something that a lot of bacteria might want. And if you use that it's kind of representative of like kind of walking down the axis, finding kind of like a short path to that particular biomass region. that reproduces experimental fluxes in E. coli very well. So it has been shown that if you use parsimonious solution, it's actually really good. It's even better than, for instance, incorporating transcriptional data, like to validate that. And it's like kind of based on that assumption that bacteria do not like to produce more enzymes than necessary. So what does Microbiome do? Well, Microbiome is basically an extension of flux balance analysis to microbial communities. That's where the name comes from. Microbiome, microbial community. So it kind of incorporates all the data that you give it. So initially, you usually need to give it at least like the abundance of the bacteria in your system. And that's a bit of a caveat there that like it actually wants it in gram dry weight, but we sequence. So what we have is more like relative abundance of the reads. So DNA extraction has a bit of a bias, and maybe some of the TAs can post Amy Willis's paper on bias there a little bit. She analyzed that really great. So there's kind of like a bias. So the DNA extraction will break down certain bacteria better than others. So you might get like more reads for one bacteria just because of that. But like the orders of magnitude are more or less correct. And you can also specify particular diets that you assume that are present. It will build up basically a huge stoichiometric matrix that does not only contain every single biochemical reaction from each organism in your sample, but also all exchanges between them and also with like the external environment, which in our case, for instance, would be the gut lumen. It can identify unique growth rates basically for each of the bacteria. And from those growth rates, it also identifies all the fluxes. So it kind of tries to reduce that even bigger flux space into like a smaller region. And then you can use stuff like parsimonious solutions to kind of go back to like a most likely flux distribution. And if you have that data, you actually get kind of a really, really detailed view in your system. So it is still like a prediction, but it's like an extremely detailed one, which will tell you every single flux and every single bacteria. And those are hundreds of thousands. So it will tell you exactly what is produced by E. coli, what is imported by E. coli, what does E. coli consume inside of itself, and how does it convert it, how much is produced into biomass, and so on. So that's like kind of a cool tool for that. And we basically have two interfaces for that. We have like a QIIME2 and a QIIME2 interface, but also a Python interface. We'll be using the Python interface, but they're almost exactly the same. Honestly, they produce the same visualization. It's the same like algorithm. And yeah, it just like the Python interface is a bit faster to install. So we'll use that just because. And we can do a little bit more with it in certain settings later on. But like most of the workflow you can do with either. So yeah, feel free to use either in your daily work pipeline. Okay, but okay. Now I rambled on way too long. Well, not way too long, a little bit. So let's actually do something, right? So the first thing we will do is run some setups. There's one thing that Microbiome actually, so it solves really large numerical optimization problems. And there's actually two different ones it solves, quadratic and linear ones. And it's really hard to find solvers that can do that well. There's obviously a few commercial ones. So unfortunately, those kind of problems also rise a lot in industry. They're very valuable to a lot of companies. So the algorithms or the programs that do so can be solved pretty expensive. So IBM has one and Gurobi also has one. And usually they're very expensive, but they're actually free for academic use. So you can actually just apply if you're a student and you will get it for free. We actually use an open source one that's pretty recent, which is OSQP. It's a great solver for quadratic programming problems. It's as good as the commercial ones always. It's a bit weaker with linear problems, which we also will solve with quite a few of, but it will work for our tutorial. The only thing I have to mention is like, we will lower the accuracy a little bit. So like the numerical accuracy, since all of those are numerical algorithms, like 10 to the minus 3, which kind of means if something is smaller than 10 to the minus 3, we cannot really say if it's really different from 0. So we kind of have to be careful. If you use a commercial solver, that can be lowered. But yeah, it's just kind of a cutoff where we say, like, if a flux is 10 to the minus 4, it might actually not even be happening. So we will start by kind of loading in the material again. Oh, sorry. You might want to do your own copy again. So you can go on File, save a copy and drive again. Rename it if you want. and then we run it. So basically you can start with the first one. If you copy it to your own drive, it will be trusted. You can also run it directly, just nothing will be saved. So that might not be super convenient, but yeah, it's possible. So that will again download all the materials. We'll also download actually the last abundance tables that we had, because that's what we're going to use. We're actually going to use exactly the data that we generated in the first tutorial here to actually connect it with Micom. For Micom, usually you can just install Micom with pip, for instance, if you use Python. It's like super simple. In this case, we do actually do quite some extra steps to get OSQP support. That will be official in Micom pretty soon as it gets into Cobra Pi. But right now we kind of will install some development versions, so that looks, you actually don't have to like really look through that in that much detail, because that's nothing in the future that won't be necessary anymore. And it's basically just an arrangement that we can run that course with open source software. And that's what all that does. So basically that like pulls in the latest development versions of the OSQP support, so we can actually use that solver here. So we run through all of those. And then in the last step, we also want to enable the QIIME2 interactions. And since these tables are like kind of saved in the biome format, we will install that as well. That will give us access to the biome format and reading those tables that QIIME generates. And that's all we need to do here to install it. Now we have Micom set up. It will run just fine. We'll take a few seconds here, but yeah, we're done. So that should all work. So you can go through the entire setup session, basically down to the big parts that says Micom. And just to reiterate, that's what we kind of want to do today. So we will start out with our raw abundance data. We will need a model database. Like I said, like since we map SAT16S data to metabolic models of the entire organism that contains like all of the biochemical reactions, we kind of need that from somewhere, right? So we could map it to existing genomes and then like reconstruct metabolic models, but that might be pretty costly. We actually provide a lot of pre-built databases. The ones we're using today has about 800 strains in there. And then if we have that, the abundance in the database, we can actually build up the entire model, and then we can start simulating growth basically. So we will basically find the fluxes that maximize growth and do some tricks to get that really well and get all the fluxes as results. Okay. To kind of see what is the input data that Micom actually wants. So how does that look? And to kind of get an idea, we will actually load in some example data that comes with Micom. It's a really constructed example where we have like 4 different E. coli strains, and we kind of want to see how that looks. And basically what we need actually is, for the abundance table, you kind of combine all the abundance and taxonomy information in a single table. So now like you have basically a column where you will say, okay, what's the genus, what's the abundance, and which sample? It's kind of the combination of both and kind of a long data format. The file comma is actually just an example. It's not required if you use a database, which was like before in the initial days of Micom, it didn't actually use databases. It built up all the models on the fly, which actually is slower. But yeah, but the thing is, okay, that's not exactly what we got from QIIME, right? We got QIIME artifacts, and taxonomy was a separate artifact from the abundance. So that looks like we have to do some work, but actually we don't. We can actually just do it. Because Micom actually has an import function for that. And before we actually get there, we will download like the model database, and we will use like the Agora database that comes from Ines Thiele's lab, and Ines will actually give a talk in our symposium tomorrow. So you will be hearing from the very experts on that topic, where they really painstakingly, with a lot of work, reconstructed metabolic models that were actually manually curated. So they reconstructed by now for about 800 bacterial strains, metabolic models, but then actually went and validated them. For instance, they checked, oh, if the metabolic model predicts that you can grow on that growth medium, does that actually work? And then they went back to create the model. So they actually did reproduce like the actual phenotype. And they, for instance, they checked if that organism is known to be able, for instance, to assimilate nitrogen. Is that actually true? Things like that, for instance. So So they actually like defined also metabolic functions that must be compatible. So it's like a really well curated database, and Agora is kind of specialized for the gut microbiome. So the strains contained in there are the most common ones that are found in the human gut. So it's perfect for our data, right? So we will download that, and we will download that on the genus level. So those models are already summarized on the genus level because 16S usually gives us like good information down to the genus. Species is usually really hard. So that's kind of the level we will use. So we'll download that database. It's a little bit bigger, still should be fairly quick. And now we have our model database. And now we will also import the data. And that's in the treasure chest folder. That's the same data that you actually generated in the last tutorial. What we will do is basically, we just import the QIIME data. You actually don't need QIIME to install to import QIIME data. So for instance, you can get that from a collaborator that has used QIIME and just use it with MyCom without even installing QIIME. It knows how to manage that. And you can also say what if you want to collapse it. And since our model database will be genus level, and that's kind of the best taxonomy information we get from 16S, we will also collapse on the genus level. What it will basically do is it just takes those tables and builds up that table that we actually need, like the taxonomy table for MyCom. Okay, that will run. You will not really see any output, so don't be too surprised. If you actually run the next cell, it will actually show you what that looks like. And if we look at it, it's exactly what we wanted. So we have a sample ID. We have the abundance, which is just the number of reads of that particular genus in that sample. We have the name of the genus, for instance, Akkermansia here, Bacteroides here, and an ID, which in this case is the same as the genus. You can actually like build up rules to make the IDs more informative. Like sometimes you have like weird names that have spaces and you want to substitute with underscores or something, but yeah, for now it does it automatically. And also gives us a relative abundance. That's kind of the proportion. So for instance, we see that there's already quite some differences. So that sample has like not even 1% of Akkermansia, but that sample has 82% of Akkermansia. So that's a huge difference in here, for instance. And maybe just to comment a little bit on those levels. So if you give data to MyCom, usually there's like several ways how it can later match it to a database. For instance, you see here that it only has a genus column. It could also have columns, for instance, for the phylum, the domain, like for the phylum, the kingdom, the family, the class, and order. The more columns it has, the more it will also use to match it to the database. It will always require that all taxonomic ranks in this table are also exactly the same in the database. That can sometimes be a bit of a problem because different databases use different systems for that. So for instance, Green Genes might call it Prevotella, and then like Agora might call it something different. It doesn't, but sometimes that can happen, even though the species is the same. So there can be less matching to the database if you assume that the entire taxonomy has to line up. So that can be like a small problem, but we will actually only match on the genus name right now, which will work for our data set. That might actually be bad if we have like two different organisms that happen to have the same genus name, but in a different family. That's definitely valid in the taxonomy. It doesn't happen very often, but it can. And then we would mismatch. So it's a good idea to kind of later kind of check that it did it well. But for now, it will work. Okay. What we can also do is we can read in our metadata from before, and we will kind of just merge that into with our taxonomy table. So here we use pandas, which is like a Python package to manage like data frames in general. And we will kind of merge those two on the sample ID. And basically that gives us a bit of an expanded table where we now have the disease status as well, make it a bit easier for us to observe what's happening. Okay, perfect. And the first step we will do, and that will be fairly quick, is we will actually build the community models. It still takes a bit of time. They're actually not small. We will look at some in a few seconds. We'll try to get something that actually, represents a full model of the microbiome community for each sample. It's actually not that hard. So that's like normal Python code here. So basically we import from MyCom, we import the build workflow. We could also import community. We're not really going to use it, and pandas, but we don't need it here. So sorry about that. We could even kind of remove that here. And then what do we need to build the models? Well, obviously we need our taxonomy information. We need a model database, and that's the one we downloaded. we need a folder, and in that folder it will basically save one full microbiota community metabolic model for each sample that you have. We have to specify the solver. In this case, we specify OSQP. Then we can specify a cutoff. So we can say if something is less than 1%, for instance, in the abundance, we kind of ignore it. The logic here is that if something is very, very, very low abundant, it cannot really produce huge amounts of metabolites. There's like certain thermodynamic levels, like a single bacteria can simply not produce like micromolar concentrations in the gut. It's really hard. It would need to kind of really go against physics. So we can kind of use that to exclude it and make the models a bit smaller if we want to. We will be fairly aggressive here. We would say that everything that's like less than 2.5% abundance, so that's like a fraction. So percent is like a factor of 100, so that's 2.5% here. We will remove those, so we kind of stay with only the most abundant thing, and that's mostly here to be a bit faster with all the simulations that we do. In general, we actually do 0.01% or even less than that, so we really only remove things that are really, really low abundant. And we use 2 threads that will automatically parallelize everything across the models for you and show your progress bar also and how that's going. And that will kind of do its course here. And while that is running, let's go back and actually understand what's happening. So what does Microbiome actually do that makes it a bit easier to get flux distributions? So like I said, like usually we can kind of look for the maximum biomass, and often that's quite good because maximum biomass for a single bacteria is usually a single value. So that's great. But what does growth actually mean for community, right? If it's usually we talk about growth rate for single bacteria, does a microbiome, like a community, also have a growth rate? And it does. You could measure the OD of a bacterial community of different bacteria, for instance, in the labs, the same way you measured for a single bacteria. And that overall growth rate for the entire microbiome community is usually expressed by a community growth rate, and I will call that mu C. Like mu is usually what's used for growth rates and C for community. And it's given basically like as a weighted sum of the individual growth rates, and it's weighted by the relative abundance. Because, for instance, if you have 2 strains in there and both grow with a growth rate of 1 per hour, and they're both in the same abundance, and on average, this entire thing community all still grows by the growth rate of 1 per hour. But if one was twice as fast and more abundant, it would kind of be weighted more, like it would contribute more to the overall biomass that's produced. So that's pretty straightforward. So we could think, okay, well, we extend it in a way like we did for a single genome scale metabolic model. We just now maximize the community growth rate, right? But well, there's a little problem here. The thing is, there's infinitely many solutions for individual growth rates that are actually consistent with that. And we can build up an example here. Let's assume we have really simple example, almost stupid. Like we have 2 E. coli strains in here, exactly the same abundance, both 50% in the community. And we already know that maybe the maximum community growth rate is 1. It's exactly the example that I just made, for instance. So kind of both of those growth rates weighted by the relative abundance has to be 1. But there's a lot of possibilities, right? So it could be that this one grows with a rate of 2, then it would be 0.5 multiplied by a growth rate of 2. And the first one doesn't grow at all. Average would be 1, that works. The other, we can turn it around. That one grows with a growth rate of 2, also works. And actually, if we kind of plot both of those possible growth rates, everything that lies on that line is completely feasible. Like all of that is fine. That all of that gives us the maximum community growth rates. And that's like not good, because now we get like ambiguity, and that basically means that our flux cone becomes way bigger. Like it's actually harder to identify those individual growth rates now. Like since we have like all of them, there's infinitely many just on that line, right? So that's not what we want. Intuitively, we could say, okay, and that's what we started off when we developed Microbiome. We kind of said, okay, if there's exactly like similar strains and then the same abundance, we know nothing else, we kind of would assume they grow on the same rate, right? It's the same organism. So we tried out like certain methods, what would give us that result? That was actually like the motivation. And we actually showed that what you can do is a method of regularization. The idea of regularization, we actually saw that today in the parsimonious FBA. It means I take additional data or additional information about the biological system that will help me to reduce the solution to like a smaller subspace. And the information that we took in is that we said, well, like if you measure that a particular organism is present in your system, it has to grow. So it should not have a zero growth rate, but it's kind of intuitive, I would say. And we checked which regularization method would kind of work. And we proved that actually no linear method can ensure that it's actually true. If your abundance data shows that it is not, well, it like cannot be consistent with your abundance data, but a quadratic term actually does. So what you can do is you can minimize the sum of squares of the growth rates. And if you do so, it will try to it's like seems weird, why would we do that? But like an intuitive representation is it tries to actually distribute growth over as many organisms in the system as possible. For instance, for our example, it actually ends up with the point where it says, okay, both grow at the growth rate of 1, exactly the same, and the community growth rate is still 1. It reduces that infinite solution space and growth rate to a single point. And then we kind of also observed that you can also have the case like, basically, those bacteria still compete, right? So a single bacteria might not be too happy to actually sacrifice its own growth completely, basically to die off just to maximize the community growth rate. So often what you get is like some suboptimal community growth. You might not actually hit the maximum growth of the entire microbiome community. You hit something that's a little bit smaller, but ensures that everybody can grow because they're still competing for resources and want to survive. So that's basically, you can do that and you basically say, okay, I don't want to reach maybe the maximum growth rate, but another one. In that case, it kind of gives you the same thing. So if you say 80% optimum growth, you kind of get like a little bit slower growth rates. And that can kind of like really affect that. So that's basically what Microbiome uses. But that's kind of, okay, I argumented here with a lot of like maybes and stuff. So you might actually ask if that works. And that method was actually called cooperative trade-off FBA. Cooperative because that regularization basically ensures that everybody can grow, which is kind of cooperative. And trade-off because you make a trade-off on the maximum growth rate. So that basically allows us to treat metagenome scale models with the same methods as genome scale metabolic models because it gives us a unique growth rate for every single organism in the system. And that's really similar to normal FBA. So then I can use parsimonious FBA and minimal media, all those cool methods that like FBA usually allow us without having to deal with that vastly increased like flux cone. But does it work? That's the main question. So the first thing is like I claimed that if we do that regularization, we get growth for more taxa. So that's the first thing that we looked at. So that's basically the density distribution of growth rates. And you see if we do nothing, if we just maximize community growth rate and take whatever solution the solver gives you, like what will happen is that the numerical solver will give you some solution that's easy for it to get, that definitely lies in that huge flux cone. But is it something that seems reasonable? Well, what we observed is that very few taxa, in most cases only a single taxa would grow at a really high growth rate. Everything else 10 to the minus 15, essentially absence of growth. It's not even there. So there was nothing. If we maintained the maximum growth rate and introduced just that regularization, we saw a massive shift already. Like you see now that suddenly a lot more taxa can actually grow. And if you actually like pull down the trade-off, it's really easy to get into a case where all of the observed taxa in the system can grow. And that is consistent with the 16S data. We observe those taxa, they live in the gut, they're able to grow, and they can grow, but you will not find those solutions if you just like don't do like the corporate trade-off. And then we also try to validate that. So since Microbiome basically selects growth rates, like one of the obvious things was to see if those growth rates like kind of correspond to what we have. But how do you get growth rates for bacteria growing in the gut? Well, in that particular data set, we actually use metagenome samples where we get the entire genome. And bacterial genomes usually replicate from one origin of replication, and then the circular genome gets kind of replicated completely. So if you map the reads and you count how many reads map to a certain area on the genome, that's often called the coverage profile. It tells you like how much of that genome is covered at what rate at particular points. And what you usually see is that obviously since it starts replicating of the origin, you have like more reads there. and you kind of see that decay like to the end of the genome. So, and you can use that slope basically to estimate the replication rate. And that was a science paper from the Seagal lab. Maybe CTAs can post that in the channel. And they have actually shown in that paper that it actually does reproduce the actual growth rate. This is replication rate. It's not directly growth rate, but often those are kind of have to be aligned, right? If you like duplicate and you haven't duplicated your genome, you're in trouble, right? So and what we saw is like we kind of looked at what trade-off would actually give us the best agreement. And we saw that if we don't do any regularization and let's take like that random solution that we get from the solver, it actually doesn't reproduce growth rates really well. And then if we still assume maximum community growth, it doesn't really get that much better. But then like at the trade-off here starting at 0.7, we actually identified 0.5 was like the optimum point here. We actually do get like a positive correlation with the replication rates that we got from the metagenome data. So obviously it's not perfect. One thing is that here we kind of didn't have any diet data. So we didn't really know what metabolites are present in the gut to be eaten by bacteria. So we assumed like an average Western diet for every individual in that study. And that was obviously not true because people do eat differently. So we wouldn't have expected perfect agreement, but we would have like expected some agreement. And we see that really big difference here of using that method over like just naively optimizing community growth rate. So it kind of worked. So easy peasy. So what's taking so long then usually? Well, metagenome scale models are really big. So that's kind of what, for instance, a realistic one looks like that we usually have. So what I kind of plotted here is that everything is a metabolite, and blue points here are reactions, and if they're connected, basically a metabolite gets consumed by a particular reaction. You can't even see the points because there's so many of them. In total, we have like 70,000 biochemical reactions going on. There's 47,000 metabolites. It's so many metabolites because we consider metabolite glucose inside of E. coli something different than glucose inside of Bacteroides, for instance. And there's like 300,000 connections between them. So it's pretty massive what has to be modeled. And you kind of see in that visualization, actually, the kind of really interconnected parts are actually the individual genera. For instance, here you kind of see imports from the outside, and then like the kind of empty space in the middle, pretty much, those are exchanges between microbes where they exchange metabolites. So that's like a huge problem to solve in mathematical terms. So if we go back to our notebook, it's actually done building it, and it will give us an output, which is a manifest. And that's really good to do some checks right now. So basically tells us, okay, for that sample in the database, I found 5 of the 5 taxa that you actually still had left in your model. Since we had that really aggressive cutoff, we're left with kind of not that many genera, and tells you also how much of the taxa we identified. And we see in the worst case, we still find 9 out of the 11 taxa in the database. There might be some that we can't find. So that's usually pretty good. And it also will calculate it in abundance because that's only like looking at the taxa. But for instance, a taxa that's really, really low abundant, you might be okay not being found in the database compared to like the most abundant taxon. So you kind of want to have that expressed also in abundance. And here it tells you that at least 80% usually of the overall abundance in our sample is also represented by the database. So that looks good. Like usually we say more than 50 is even okay. If it's less than 50, obviously you're not representing the actual biological system at all anymore. So that would be problematic. So to kind of start growth, like I said, we actually need to give information about which metabolites can be consumed, which is very different from other methods. And I think it's actually a plus here that you have to be explicit and say, okay, I assume there are starches actually that can be consumed. And what we will load in is the average Western diet. Since we know nothing about those individuals in their diet, we can kind of assume, since they are on the Western continents, that they might have eaten an average diet. So let's just assume that. We can load that in, it will download it. And that's actually what we will use as a growth medium. And let's look how that actually looks like. So we can, again, use load chime medium from the microbiome formats part. you just put in the file name and it will tell you what it looks like. So what we see now is we see a whole bunch of reactions here, which are the input, the fluxes for the export and import reactions. In this case, it only signifies imports. The flux is the actual rate by which something can be imported at its maximal rate. It's a bound. It's not the actual flux. So as we saw before, we have those like flux bounds on each flux that usually tell us if often those are just thermodynamic constraints. You make them really big. We just say, okay, like you can't be faster than diffusion, for instance. In this case, it's kind of based on the abundance of those things in an average diet, kind of calculated over 24 hours. We also have like a dilution thing. So what we did in the paper is actually, since most of the stuff gets absorbed in the small intestine, it doesn't actually get into the large intestine that easily. So we kind of depleted those as well. And we assumed that like 90% of that gets removed in the small intestine. And you actually saw the metabolites that are related to this. For instance, Fru-M is fructose and GLC-M is glucose. That's kind of the amounts that we have in here. It's really, we actually don't get a lot of glucose in the large intestine because it gets absorbed in the small intestine. So it's kind of adjusted for that. And with that diet, we actually start simulating growth. So we had the growth results. That's what we want to get. Our manifest is the output from before. It kind of just tells us what models and what samples we have. We have a model folder. That's exactly the same that we used before to save the models in. So that folder, and we can look at it. If we go to materials, we have that models folder. We see those weird pickle files, and that's literally what it is. It's like a canned model. So they're all in, like, and we have one for each sample and the manifest as well, if you want to look at that. So we have to specify a trade-off value, right? So if we go back to what I showed you in the validation, we did it actually with actual replication rates, and then we chose the trade-off that actually works out the best. But we did observe that you don't need to do that. Usually that one coincides pretty much with the one that allows growth for the most taxa. So if you wanted to choose that, you can just go through, test a few different trade-off values and use the one that allows growth for hopefully all taxa. But also we observed that 0.5 usually works pretty well. So we can kind of start with that. And it's the same, we can give it 2 threads because we have 2 CPUs. And we also save that using a Python library called Pickle, which will kind of compress the output and save it for later use in case you want to use it. Exactly. That's a cool Pickle GIF. So that will take about 3 minutes now. So yeah, that shouldn't be too long. In case you don't want to run that, you don't want to wait, you can also run the second one. So if you don't run that one, the next comment here will actually detect if that growth results have been generated. If they haven't generated, we'll just load like a cached version, but it's not taking too long. You usually also get like that indicator here. So it tells, oh, I took 30 seconds for the first model. I approximate that I will need more, maybe like 3 minutes more to go through all of them. So if you have more cores, more CPUs, that will run across as many as you want. So actually, since it kind of processes every single sample individually, that parallelizes really well, at least with many samples. So if you have 100 samples, you can run that on 100 cores if you want, and will be really fast. Like it will be done maybe in a few minutes, even for large models. So it's really not that bad. You need some memory for that. So a good rule of thumb, if you want to run it on real models where you like shows a cutoff, like abundance cutoff that's actually lower, like 0.1%, is like 1 gigabyte per model. That works really well. So if you want to process 100 samples in parallel, then you will need about 100 gigabytes of RAM as well. But if you have a computer that has 100 CPUs, it probably has 100 gigabyte of RAM as well. That would be like already a massive computation server. But yeah, it does parallelize really well. It's just a bit slow here because we don't have that many CPUs. But I mean, we're halfway through just me babbling and probably will be done soon. But maybe let's already kind of see what we will get out of that. So that simulates growth. And what it actually does is it actually uses the same concept of parsimoniety, but it mostly uses it for imports. So in the gut, bacteria compete with our intestine for resources, right? We have a huge surface area of intestine in there that just absorbs metabolites like crazy if it can. So if you are a bacteria there and you want glucose, for instance, you have to be faster than that. So you have to be really quick in absorbing it. And that kind of means you want to use the smallest amount possible, because if you can grow on less glucose, then it's cheaper for you to get what you need to get maximum growth. So it's the same concept of parsimoniety, like bacteria like to be efficient. Why should you import more glucose than you need for maximum growth? It doesn't really make sense. So it will do that as well for each model, and that will actually pinpoint the export fluxes well better than just not doing that. So what you will get is actually 3 sub-data sets in the growth results. One, you obviously get the predicted growth rate for all taxa and all samples. That often correlates a lot with abundance, so it's informative. Like, it tells you if, for instance, if you have inconsistencies, if you see that something is predicted to not be able to grow even in low trade-off values, that often means that your assumption about the diet might be wrong. It also gives you import and export fluxes for each taxon and the external environment. So that's really interesting. It will tell you which part of the diet is actually preferentially taken up by that particular sample. Even if we have the same diet for each sample, one of them might, for instance, require more starch than another, and you will see that. And it will also give us annotation for fluxes. So that's quite cool. So let's look at that. In my case, it finished, and it should finish for you pretty soon as well, I hope. So let's look at the growth rates. How does that look? So we basically see the genus, we see the abundance, and then we also see the predicted growth rate. That unit of the growth rate is 1 per hour. We see also the number of metabolic reactions in that particular genus, the number of metabolites, the trade-offs that we used, and the sample ID. So we kind of know that Bacteroides in that sample grows at that rate. It's already cool, but I think like the most interesting part are the exchanges for me. So you can kind of see that, for instance, Faecalibacterium in that particular sample produced, for instance, that particular metabolite, and we will look at how to actually identify it at that rate. And basically, if it's positive, it usually means it gets exported or produced, and if it's negative, it means consumed, for instance. So you kind of know what it like consumes and produces, and like it's a very detailed view. You see that you have 31,000 exchange fluxes here, just that you get. This is not the internal fluxes that happen inside of the bacteria. It's just exchanges from a bacteria with its environment. And that's already like kind of very detailed view of the system. And it also locks the tolerance for you, so you kind of know when to ignore a certain flux because it's too small. But like, how could we actually tell what that is? Like, that's not really super informative here. Even if it tells me the metabolite is 1, 2D, well, I don't know what that is, right? So for instance, we might be interested in metabolites that's called AC. Well, what is that? Is that air conditioning maybe? That's quite some feature for a bacteria, but well, let's look it up. See, one thing that the growth results also contain is an annotation, and that's like quite helpful to map it also to external databases. So essentially that's the pandas data frame, and you can look up like a individual name, and it will tell you, okay, what is that? Well, the metabolite name, okay, we know that it's AC. Oh, it's acetate. It's not air conditioning. Okay, makes sense. You also get like cross-links to certain databases. For instance, the human metabolite database. You actually get an INCHI screen. That's basically like a short representation of the chemical formula. You get a KEC compound number if you want to look it up in KEC. You have a PubChem compound number, and you get the reaction that actually imports it. So that might be helpful. What you can also do is, if you want even more information, Agora has that really cool website, and I'll show you how that looks. Let's open that in a new tab, which is called the Virtual Metabolic Human, and Ines will probably talk about that tomorrow. So I'm not gonna take that away, but if you go and start, you can basically go here on home and say, Map Navigator, browse gut microbiota. And you can look for species, but you can also look for metabolites. So for instance, if I enter AC here, oops, maybe might have to scroll a little bit to find it. You can also use acetate here. You find the same thing. It says it's acetic acid, and you actually get even way more information. You get a full description, a full text, what it is. You get a lot of additional databases. And what's also kind of fun is you can actually look at which bacteria produce it. So you can, for instance, say present in which microbe reaction. That tells you every single biochemical reaction where that is used in a microbe. You can also say in which microbes it's present, and we see it's basically present in all of them. So Agora right now is 818 strains, and every single one knows acetate, so it's always there. You can also see where it's a fermentation product, and yeah, you can even look into human reactions that have it. So if you want to see, oh, how could that affect a human? Let's see, oh, okay, those are all the enzymes that humans have where it's actually acetate is involved. So you can get a lot of information about that back by that. And yeah, we have our growth data. So let's actually look what we got, right? The first thing we want to actually look at is the growth rates a little bit. So in Micom, we can generate visualizations that are really similar to QIIME visualizations, only that we don't need to use the QIIME viewer. It's just like an HTML file, essentially a website. You can download and open that with a browser, or often it allows you to open it directly in your browser. So for instance, the first one is we can use the plot growth function from the Micom Viz sub package and kind of show the growth results. So if we do that, we actually get a file generated here, which looks like that. So you get growth rates and then the current date, and it ends in HTML. So it's a web page. You can download that. And if you have it, you might want to open up with your browser if you can. And if you do that, it basically shows you what it describes. So you basically see each point is a single sample, and you see the growth rate of each of the genera in that sample. If you click on a single point, it will show you only the growth rate for that particular sample. So for instance, what we see here globally, and you can double click in an empty space to kind of reset it, a Bacteroides seems to be the fastest grower in that system. And it's followed actually by Fusobacterium. And Fusobacterium isn't really present that often, but it is a bit, it does grow fast in the sample where it's present. So that kind of means that it's expanding really quickly. And sometimes you see that separation as well. And you could, for instance, try to track which ones are healthy and which ones are sick. But yeah, basically it will rank like and show you which are the fastest and slowest growers in the system. That's okay. It's informative, but we might be more interested in what we can do with the exchange rates, right? So one thing is to look at growth niches. The first thing we could do is just check every single sample. If you have questions, I think the Slack might be better if you have access to that, because I can't see the chat in Zoom right now when I'm presenting my slides. So go ahead and people will help you for sure. So the first thing we can try to ask is if we can actually just see like what is preferentially consumed in every single sample. And that's basically the plot exchanges per sample. So if we do that, same as before, we get like another HTML page in here, one that's called sample exchanges. And if you download that one, we open this one, it looks kind of like that. So it will basically show us for every single metabolite in that system. And if you hover over, it actually gives you the full name of the metabolite. Yeah. Oh, you can see. Oh, yeah, because the different window. I'm so sorry. Thanks for that. Let me try to rectify this. I will stop sharing my machine for a second and try to actually share my entire screen. This should be the one. Okay. Can you see it now? Yeah, perfect. Thanks so much. And thanks for raising your hand and bringing that up. I will actually, I think, yeah, you can just look at the growth rates yourself as well. So that kind of shows you the metabolite utilization. So we assumed the same diet for every sample, but that doesn't really mean that much of the particular metabolite actually gets consumed. That's just an upper bound. We actually see, for instance, that here for AMP, adenosine monophosphate, we see it's like, for instance, only imported in a few of the samples, but not in others. And there's a few more of those. So you get the entire profile here and the samples on the bottom. They actually grouped like so that similar samples are next to each other. So we do see quite some heterogeneity here. So you do see, even if you give like different microbiomes, the same diet, they will eat different things because it's different bacteria, they have different preferences. And what also is like really interesting is to look at what individual microbes consume, right? So that goes into really the idea of a growth niche. So basically we want to see is a certain bacteria that eat similar things as other bacteria. So do they overlap in their consumption of particular metabolites? And that can be done by the exchanges per taxon. And what that does is actually, since it's really high dimensional, like what each bacteria eats is a lot of different fluxes. What it will do is it basically does a 2D plot with a similar idea as the embedding that we saw in the first tutorial. It's the same idea. Basically, it will arrange bacteria close to each other if they consume similar metabolites, and it will not do it if they consume very different metabolites. So those will be far together. And it uses a method called t-SNE. That is really good at doing that, basically. So if we generate that, you get like another plot, and this one is now called taxon exchanges. So you can do the same thing, download this file, just open it up directly. Let me move this around. And this looks something like that. Like I said, it's like a projection. Essentially, if you're in the middle of that map, that means like you're kind of consuming similar things. And if you do that, you can also just click on one and you get only a single genus. So you kind of see that usually they kind of occupy similar growth niches. For instance, all the Bacteroides kind of are close to each other in each sample. But they might actually overlap with others like Lactobacillus here. You can double click again to set it back. And you do see there might be outliers. So there's one Coprococcus that kind of consumes very different things here. And this can be quite interesting, especially if you're looking for like a particular organism. You want to see if it, for instance, competes with a lot of different bacteria, or is it really its own thing, or does it adapt and kind of move its preference a little bit? So let's see if we can maybe find Clostridium here. One thing about Clostridium is that we see it does differentiate a little bit. And that's if that pathogen is present, like that might include C. diff. We actually don't know. Clostridium is a pretty large group, but we see it kind of does compete with a lot of different microbes here. So there's certain other microbes that are close to it. And that might be like a reason why it kind of prefers maybe, for instance, to be kind of more isolated, because that kind of eliminates competition. And it can definitely get higher growth values. And yeah, maybe the last thing we might be very interested in is metabolic connections to a phenotype. But maybe before I do that, I'll just show you what we found for that later data set. So basically, when we looked at the same plots that I showed you for like a large data set of like healthy and diabetic individuals, and we first looked at like the consumption preferences, we actually found that independent of whether the individuals were sick or not, there actually is like kind of almost a bimodality. Where kind of the microbiome either prefers to eat off more fibers and starches, or it prefers to eat more branched chain amino acids and amino acids in general. So there was kind of that preference switch where a few samples seem to kind of like to eat more protein and some others like more fibers and starches. And then when we actually did like the same niche plot there, we actually saw that mostly everybody stays within their lane. Like all of the genera kind of occupy a very distinct point in the niche space, which kind of makes sense because we know that like the best thing you can do if you get into a new ecosystem is actually find an unoccupied niche. So you go into a space where you're not competing with anybody. But we did see that there's like definitely areas in the center where things at least get close. And there's some interesting ones that we saw here where like things that often like you don't see together kind of also seem to prefer similar growth niches. Okay. And yeah, when we looked at basically, we actually used the same thing. So when we looked at those export rates, for instance, and overall production of butyrate, the capacity, for instance, we saw that as it has been observed, like in real measurements, like we saw that short chain fatty acids, which are some compounds that are really important for the gut, they're produced by microbes and they usually help to differentiate our intestinal cells and also keeps them healthy. They have some differences, like for instance, we saw there was less butyrate production in type 2 type 1 diabetic individuals compared to controls. And we could actually also look at which bacteria produce those short chain fatty acids. One thing we observed is that for a lot of them, actually had quite a huge spread. So depending on the sample, actually a different genus might take it up. It's like they all kind of, for instance, produce more butyrate, but then like not the same genera necessarily do it. And that I think is like, yeah, that was important because like a lot of times, like when we do taxonomy analysis, we do things like saying, oh, Roseburia is a butyrate producer, so that's why it's beneficial. But actually Roseburia might or might not produce butyrate depending on the diets that you give it and which other bacteria that's there. So that has to be taken into account here. And yeah, we can actually use that to try to find connections to a phenotype. So what we can do is we can look at the production rates, and I have that here, I think. So what we often look at is the overproduction flux. So there's two things we could look at. We could look at like the excess production. So basically, there are metabolites that get exported into extracellular space in the gut, and then they get not taken up by other bacteria. And then there's like an excess of it, and you could look at that. But I think more important is like the over rate of a certain metabolite that it gets exported in the extracellular space, because even if it gets taken up by other bacteria, that is the pool that usually competes with intestinal cells. And one thing you might have noticed is that Microbiome right now does not have a model for an intestinal cell in there. So there's no real competition for the microbes with intestine. So usually, and you can show that with the balance equations as well, but maybe you just believe me, that this is the pool that the intestine would actually have access to. So that's often really, we saw that it in general is more related to the phenotype. So that's like, for instance, the overall rate of butyrate that's produced by bacteria in the system. And that is actually what we can use as well to get a connection with the phenotype in the next part. And basically what it will ask you to do is, again, you need to pass in the phenotype that you're interested in. What we can do is we already saved the manifest data and read it. So we can basically just, we will read it, we will set the index, that's not really important, but then we kind of choose again the disease stat column basically as our phenotype. So we just make a plot for that. We use the growth results, the phenotype, we define a tolerance, and we will use the 10 to the minus 3, and we say we want production fluxes. And if we do that, we'll actually fit a logistic regression. Don't worry about the warnings, that's all fine, nothing burned here. And it will actually try to find fluxes that are predictive of the particular phenotype. But now it's not really bacteria. It will tell you exactly, okay, the microbiome community produces that particular metabolite, and that will give you will predict actually the phenotype. And if you run that, it will generate that fit file here, fit 2020, which is like the logistic regression model. You can open that up. And what does it give you? The first thing is it tells you only the coefficients that were actually predictive, basically coefficients that were above a certain threshold. And you could even download the data. That works for all visualizations actually that are generated. You can always download the raw data underlying each plot. And we can look what we get here. So we have law rate here, and that has a negative coefficient. A negative coefficient means associated with the first group. The first group is recurrent C. diff infection in that case, because it starts with a capital R. So that's like the, the alphabetic order that it uses. So it says like law rate here is associated with recurrent C. diff infection. Okay, we might have to look what that is. Pyruvate. Okay, that's like we know that this is something that gets produced in glycolysis, but also somehow related to growth rates. We see a little bit of aspartate, tetradecanoate, that's like a fatty acid, and maltotriose. So let's maybe in a second look what that might mean. And you can actually go and compare it for each of them. For instance, if you want to compare it for aspartate, it will show you the levels. That's law rates, the first one, and we can definitely see that this flux seems to be slightly higher, the overproduction flux, in recurrent C. diff infection. And you can go through maltotriose as well, and you will find kind of the opposite, right? It's higher in healthy individuals. And then what it actually does is it will fit models, but we'll actually try to be a bit more conservative with what we have seen in the CHIME output, for instance. So it will tell us the same thing. The training accuracy is perfect. I can, just by the export fluxes, I can perfectly predict who is healthy and who is not. But then what it also does a leave one out cross validation. So it basically trains the entire regression, but leaving out a single individual. And then it tries to predict that individual and will tell you how well it does. And we see that actually it's lower. It's like 75% accuracy. And that's kind of what we would expect, because we know that one looks pretty similar to the healthy ones, but it has a huge variation. So there's a few of them you can predict really well, and some you cannot predict really well. And it actually shows you also like that confusion plot where you kind of see, okay, of the recurrent C. diff infection ones, how were they predicted in that leave one out cross validation? You see that 3 out of the 4 were predicted of being actually sick as they are, whereas like 1 of them was predicted as healthy. And we know probably by now which one that was. And we actually see that for healthy, we also do that mistake at least once. So it tries to be conservative about having this. So that's interesting, but does that mean something? Do those metabolites mean something? Well, I put you some like fun facts here, basically. Maltotriose is a breakdown product of alpha amylase. Alpha amylase is a major enzyme that basically breaks down starches. So if the bacteria want to get access to glucose, they actually don't get glucose. Like we said, the small intestine gets rid of that, like it absorbs us for ourselves. We don't give that to bacteria. But all of the complex starches we can't digest. Those get into the large intestine. Alpha amylase breaks it down, and one of those breakdown things is maltotriose, which is kind of a sugar. Laurate, which we saw is actually super associated with, well, not super, but is associated with recurrent C. diff infection. That is interesting because it has been known as like a fatty acid that actually increases serum cholesterol more than any other fatty acids. If you eat a lot of laurate, you do increase serum cholesterol levels. And that already is like interesting here. And why is that interesting? Because I told you that C. diff is really good at detecting if other bacteria are present, but I never told you how it actually does that. It actually has a really cool mechanism how it does that. One of the things that bacteria actually break down or like to break down a lot is bile acids. Bile acids are really abundant. Like our gallbladder produces those, and we use that usually to dissolve fats, so we can actually absorb them in the small intestine. But a lot of those primary bile acids, which are the ones that the gallbladder produces, they end up in the gut. And a lot of them are conjugated with like amino acids that our bacteria love to cleave off. So they deconjugate them and convert them to secondary bile acids. And basically one mechanism for C. diff to know that it can proliferate is it detects higher primary bile acid levels. Since it knows that if you have like a really like intact microbiome community, it will usually assimilate most of the primary bile acids into secondary ones. So primary bile acid levels are low. Okay, so that's the first part of the story. Okay, so C. diff uses primary bile acid levels to detect if it can proliferate. But how do primary bile acids get produced? They get produced from cholesterol. That's like one of the major endpoints for cholesterol is primary bile acids. So if you would, in theory, increase the production of cholesterol and it would kind of end up in higher levels primary bile acids, that would actually create a feedback here, right? Like in communities that creates more cholesterol, creates more primary bile acids, that creates more proliferation of C. diff. It's a hypothesis, or we have really little data here, but it's kind of a cool story, I think. Also, like aspartate, like why do we have higher aspartate production recurrent C. diff? Well, C. diff toxins actually, which are really produced by C. diff in high abundances, are both aspartate proteases. So maybe there's some connection here, but yeah. It's very little data, but it's hypothetical. And that actually would be something very consistent to try out. So that's the thing. Like if I look at the abundance of microbes, I can generate a lot of hypotheses. But here you actually get kind of a story you can actually try out. So it says that there's some connection with starches. It tells you like laurate might be something. So that's something you can actually test, like, and then maybe see if you can use that. So it can lead to actionable stuff, but you definitely need to validate it experimentally to know if it works. Okay. Now again, it's your turn. We have a few exercises here. They should be fairly straightforward. And we think we're better in time now even. That's amazing. And basically, just to introduce that. So one thing where Microbiome also really shines, I think, and which we haven't shown so much now, is that it can actually do, you can actually run analysis on a single sample. You can really do personalized analysis. Since you actually have that working model, you can, for instance, turn some knobs. What happens if I change the diet? What happens to the growth rate of particular bacteria? What happens to exchange fluxes? That you can do for every single sample. And I kind of want to illustrate that. That's what we will do in the exercises. So there are a few parts that I did for you. We can look at the manifest again, just to see what models we generated. And if you remember, we had that weird individual, well, not weird, actually lucky individual, because, well, or unlucky, depends on your point of view. Like it was that individual here that had recurrent C. diff infections, the ends in 48, but it looked more like a healthy individual. Might mean it's like that individual is getting better, might mean it has something that even overrides those positive microbiome effects. So we might be interested in looking at that. And I'll just show you how to run everything by hand, basically. So what we could do is we can use the load pickle model to kind of open the canned model and kind of load it up. We can also open up the medium. So we load the medium again, we load the pickle, we apply the medium. It's fairly simple. You just say, oh, the medium is equal to the medium flux. Now that particular growth medium is applied to that community. And then you can start simulating. So we can start that. That doesn't really take that long. And actually tells us also, we can look at the model, it will give us some information. And it will also tell us if particular exchange reactions are missing in the model. So for instance, that particular model doesn't have imports for that. And that can be true. It's not really, it's just for you to kind of, so that warning basically exists because we sometimes saw people using like growth media specific to a model database. And if you mix them up, you might get it that not a single one of them can be found. So it's just there for that. You can look at the overall number of metabolites and also tells you which bacteria in there. So all of the information you need. You usually get 2 for each bacteria. One is like the extracellular space, one is the cytosol. In compartments. That's kind of like physical barriers that we assume are in the system, kind of, and you need transporters to get across them. And what I want you to do basically in the first one is to take that model with that applied diet. So it has that average Western diet as a single individual. So what you could, for instance, look at is in that individual person, what is like kind of the competitive landscape. And one way to get that is, well, it's a model, we can modify it as we want. So one thing we can do is we can just say, okay, I will knock out a particular taxon. For instance, I could knock out Akkermansia and see what that does to the growth rate of every other bacteria. So if it lowers the growth rate of another bacteria, that actually means there was a positive or trophic interaction. Akkermansia was providing something to the system that helped somebody else to grow. If it actually increases, that's usually commonly competition, right? If you remove that bacteria, another one can grow faster. So that means they probably competed for the same resources before. So you can look at that, and you can look at that in an individual sample. So it gives you a bit more information than just doing correlation analysis across samples, for instance. So that's what I want to see here. And you can actually go to the Microbiome documentation for that. And hopefully it's informative enough to help you. Just please note that it has that fraction argument, which is actually the trade-off value. So we use the trade-off value of 0.5. So maybe adjust that to be more realistic. And yeah, you can use that and run that here. And would be cool if we could visualize that. I think I once saw somebody plot a heat map with Seaborn in a tutorial. I wish I could remember where. That would be really helpful here, I think. And then in the second example, I kind of just want you to look at fluxes a little bit. We will actually run cooperative trade-off by hand. So I put that here for you. That will take a little bit because it actually runs PFBA on all fluxes. So you now get also internal fluxes inside of each cell, which is even more of them. You can look at the fluxes, and I want you to first identify the primary acetate producer in the system. So which organism is producing most of the acetate. And I want you to also like choose any other metabolites that you really like what's your favorite metabolite, check it out in BMH Live, see if it gets produced by anything or consumed. And also maybe try to find the metabolites that are produced at the highest level, or the fluxes basically that are the highest. Like is there certain biochemical reactions that are happening more than others? And I put in an addendum to actually show you how to choose the trade-off value with like another example. In our example here, just to take that away was not important. Any trade-off value would have worked. Since we have that little taxon there, it turns out all of them can always grow, but yeah. So for instance, if you run the corporate trade-off by hand, it kind of gives you that really detailed analysis as well. You get a community growth rate, get individual growth rates again. And it actually now gives you a fluxes argument. And you can look at that and gives you that huge matrix, but basically has a lot of 4,000 unique fluxes in each of the organisms. And you can just compare them and look at them. Yeah. And we'll see each other back here at 12:30, I think, and show you some, no, or 12:45. I don't know. Does anybody have the time plan? It should tell you in the 12:30. Oh, 2:30. Yeah, 12:30. I'm also in a different time zone apparently. So 2:30. Those should be fairly fast, so that will be fine, and I'll show you the solutions. And yeah, thanks so much. And yeah, keep the questions coming. Okay, welcome back, everybody. And let me take this off right now. And yeah, let's have a short look at the solutions for the exercises. Sorry that we did not have that on our original timetable, but yeah, we will be very quick about it. Then you still have a 15-minute break before doing the awesome talk from Noah and Tom. Yeah, and the first one I had you run like the knockout taxa function, which is kind of like a simulation where you actually knock out each individual taxon or each genus in our sample, and you see how that influences each other in a single sample. So if you run that, you kind of get that like takes about a minute, I think, since it has to run like the model several times because every single knockout has to be simulated, and then you see that heat map. And if you want, you can post in the Slack what was like your observation when you did that. So kind of remember that I told you if the value would be negative, would actually tell you that there was competition, and positive would mean that like, no, positive would be competition, sorry, negative was that there is like actually trophic interactions, so cooperativity more or less to say. And maybe just to comment on the arguments that I used like a trade-off of 0.5 because that's what we used before. And I also said that we're going to use relative change. So if you looked at the documentation, you can see that there's several ways. You could just look at the raw difference in growth rate, but since bacteria grow like in fairly different growth rates, it might make sense rather to say, oh, what percentage of growth rate like was increased or decreased. That leads to a bit nicer visualization. And then you could use seaborne as before, for instance, to use a heat map. And how do you read the output from that knockout? Every row denotes the taxon that was knocked out, and every column denotes the growth rate change in all other taxa. So if you remember, we said like negative is like cooperative, and red here is like competitive. So we see there's like some of the taxa, if you knock them out, that seem to be competing actually. And some are like very cooperative. Bacteroides seems to be very cooperative in that setting. And you actually see quite a level of cooperativity. And cooperativity is usually like harder to observe because it's a bit harder that trophic interactions take place. Competition is kind of normal. If you overlap in your growth niche, that happens. What I kind of want to point out here is like how different that is from what we often use to kind of get the same reply. So often what we could also do is if we have a lot of samples and we have like 16 S data for example, we could just look at correlations, right? We could see what's the correlation of Bacteroides with like another genus, for example, and we could build up plots that look very similar. But like, just want to point out how different that is. So one thing we see here, first, it's not symmetric. Correlations are always symmetric, but here it isn't. And I think that makes sense in ecological terms because trophic interactions are not necessarily symmetric. Like, for instance, Bacteroides could provide something to Akkermansia that helps Akkermansia grow, but Akkermansia does not necessarily provides the same thing to Bacteroides, and that's what's happening here. Like Bacteroides helps Akkermansia in that case, and not the other way around. And that can happen. Like that's absolutely possible with bacteria. It's even quite common that this happens. Like, and that you will not see if you just do normal correlation analysis. Also, the difference here is we did it in a single sample, and one thing you can actually look at is if you do that for healthy or for sick individuals, those maps will look very different. So depending on what like preference they had, which metabolites were missing in that particular community, that result might change quite a bit. So, and in the paper like that we did with Microbiome, we did that like for all samples, we saw that there's massive differences there. Like those trophic interactions depend really on the environment and who else is there, at least in those simulations. So I think it's a cool tool because you can just study what happens to the system if you knock out things in a single person, which is kind of fun, I think. And in the second part of the exercise, I had you look at like a little bit at flux data in detail. So I wanted to see what was a major acetate producer. And what we did here is basically you can just take the fluxes and find the export for acetate. So if you remember in the initial, when we did the annotation example, that was actually shown there. So if you go back here, it actually tells you for that metabolite acetate, the export reaction is named as that. So you could just have looked at that one. Oops, sorry, that's here. And then you see those fluxes. You might be a little bit unsure what that means, like in terms of if you're ever unsure what direction a certain reaction goes in, you can actually check that out by going to reactions.get by ID, and you can just copy and paste the name. And we probably get that for instance, Akkermansia, just an example. So they're actually in Microbiome internally, they're kind of always named with 2 underscores and the genus name afterwards. If you would run that, it would actually, sorry, that's the reaction, and you can get dot reaction to actually get like a string representation. And that would, for instance, look like this. So you kind of see, okay, like that's the acetate in the Akkermansia, and the product is the acetate outside of Akkermansia. And you actually see that scaling by abundance, which is something that Microbiome does and which is actually necessary to take that into account. But like what you see is the main direction of that is export. So it does export it to the outside. So positive flux is basically production and negative would be consumption. If you're unsure about that, this can be checked like such. And if we run it, so we see, now we could kind of check, so who produces most acetate, and here we see it's predicted to be Akkermansia. And Fecalibacterium as well, whereas Bacteroides, that's definitely smaller than the 10 to the minus 3 that we had as a cutoff. Okay, and then I had to kind of do the same thing, but kind of with any molecule that you like. Glucose, like I indicated, is important, so we could look at that. We see that Bacteroides seems to be the major producer of glucose in the system, and that might probably be why it's such like a positive impact on everybody else. So in this particular simulation, it predicts that Bacteroides provides glucose to a lot of different genera here. And like, even though we don't know if that would be really true in the natural system, since we did also omit a lot of genera from that simulation, it is, at least Bacteroides is really good in breaking down like complex starches and stuff. That's one of the major things that Bacteroides does, and that's what it likes to eat, like fibers, complex starches, and it breaks it down into sugars essentially. So it can definitely do that. If the impact is as big as it's predicted here, that's a question that you would have to validate. And then we could also look at the highest fluxes. So basically we can just take all the fluxes, convert them maybe to absolute values, because like they might go in different directions. We can, for instance, provide a mean, sort those, and check the first 10. And if we do that, we can just look at it, and we kind of see that the reaction here with our largest flux is a lysine transporter. You could again go back, for instance, to VMH, the VMH page, and kind of really get information what each reaction means, or go through the annotations as we've shown before. Then we have like a galactose transporter, another lysine transporter, galactose transporter. Those kind of make sense. So those are again are also breakdown products, for instance, from starches. So those are often like exchanged across the community, and certain amino acids are often exchanged. And then what we also see is like an ATPase, I think, no, ATP synthetase. So that also makes sense. Like one of the major requirements for growth is to produce ATP. So we obviously should observe some flux there. So that's what you can get. And yeah, just as an example for the trade-off, I had it here, so you could actually look at that. Like I said, for that particular example, would just tell you that every trade-off works. So I posted like an example just for like a colorectal cancer data set, how that actually looks if you make like more realistic cutoffs and stuff. That's kind of the curves that we see here. So again, you see that like fraction of taxa growing, like if you do no regularization, you get almost no taxa growing. It's like less than 20%, more around like 5 or 10. But then if you have like the cooperative trade-off method, it kind of goes up, and here you would kind of see that starting from probably 0.8 or 0.7 as a trade-off parameter, you get most of the taxa growing. So mostly how you want to choose those trade-off values is you kind of generate that curve and then look at the largest trade-off values where it allows almost all of the taxa to grow. That's like a good cutoff in most settings. But yeah, 0.5 in our case worked well as everything else. Okay, yeah, and with that, we're done. That was actually the entire course part for today. It was super fun to teach you. I really loved how active everybody was on Slack. And in case just to reiterate, in case you're doing that course like on a different time frame and you're like not doing it live, that Slack will remain online for at least a week. So go ahead and just ask us anything. If you have any problems running the notebooks, something doesn't work, or you have like conceptual questions, just ask away and we will still be responsive. Obviously, like probably more responsive in our time zone. We might not be on Slack at 3 in the morning, but oh, you never know, right? So yeah, thanks a lot. It was great. And I hope it helped you, gave you a little bit of insight what you can do with 16S data. Obviously, you could also use shotgun data, metagenomic shotgun data for MyCom. That might be even better. If you have high depths, you can even reconstruct like the specific metabolic model for the genus or the species that you have in your sample. It was a lot of fun, and yeah, see you soon, hopefully. And we'll have a 50-minute break right now, and then we'll hear like a really cool talk from Noah and Tom, who will present you like some of the insights they had when they looked at alpha diversity and metabolites and the really cool data sets that we have here at the ISB. So see ya, and yeah, keep safe, wear a mask, and keep healthy.
Hi, everyone. It's a pleasure to be here today. Oh, you see my video?
Yes.
Okay. So hi, everyone. It's a pleasure to be here today and present our work to such a worldwide attendance. I'm Noah Rappaport. I'm a senior research scientist at the Hood Price Lab for Systems Biomedicine at ISB in Seattle. And I'll be presenting today together with Tom Wilmansky, who is a postdoctoral fellow in the lab, is the real driving force behind all the microbiome analysis in the lab. We will co-present some of the work ongoing in our lab that is harnessing deep phenotyping of individuals combined with microbiome data to derive insights on health. So first, I will tell you a little bit about our lab. Our lab takes a systems biology approach to analyze human data across the lifespan. Listed here are some of the areas we're involved in, including Alzheimer's disease, studies on human longevity, pregnancy and preterm birth, and infectious diseases. What is common to all our approaches is that these are projects in which we apply computational modeling of molecular systems, and we integrate data sets that are composed of multiple data types. So today we will focus on two of these projects that are related to microbiome data together with other types of data in the areas of, and those will fall into the areas of scientific wellness, longevity, and aging. So this is the outline for our talk. First, we'll discuss the challenges of defining microbiome, what is in fact a healthy microbiome. Then we will describe the use of molecular deep phenotyping for understanding the gut microbiome. And then we will discuss the concept of plasma metabolomics as a functional readout of the human gut microbiome through a project that attempted to predict gut microbiome alpha diversity from blood metabolomics. And finally, we will present a project that studied gut microbiome in aging and longevity. So we know that the gut microbiome has multiple different biological and metabolic functions. We also know that many of these functions modulate host health. Over the last decade, so gut microbial composition has been linked to a number of human diseases, and that includes gastrointestinal diseases, metabolic diseases, autoimmune diseases, and up to neurodegenerative disorders. So there are multiple ways in which gut microbiome can affect human physiology. For example, through its effect on absorption of nutrients, its involvement in the development of the immune system, and it also has a role in metabolic homeostasis. So although we have an increasing knowledge on how gut microbiome may affect human health, there is no real understanding of what constitutes a healthy gut microbiome. So why is it so hard to determine what in fact is a healthy gut microbiome? The reason is that the abundance and the presence of specific microbes changes across different geographic locations and environments, which basically makes the translation of findings from one group of individuals to another quite problematic. So in this slide, you see results from a paper presenting data from the Guangdong province in China. This is a province that has 108 million people living in it. It's the most developed area in China with a gross domestic product in 2017 of $1.3 trillion, which is comparable to South Korea. So what you see in the slides are data for about 7,000 participants from 14 randomly selected districts in this province, color-coded by GDP per capita. The pie charts present phylum level across frequencies across the districts. At the bottom, you see the huge inter-individual variability that exists even in the phylum level. Another important result from this paper was that microbiota models that predicted metabolic diseases that were developed in one location did a pretty good job predicting disease within the data of the same location, but the model doesn't actually translate across districts when you attempt out-of-sample classification. As you see on the right hand of on the right for metabolic syndrome and type 2 diabetes, when they've tried out-of-sample prediction, the author got AUC of close to 0.5, which basically means that the model doesn't have any prediction capacity across the districts. In fact, among the phenotypes, host location showed the strongest association with microbiota variation. So the conclusion, the conclusion from this paper, which is cited here, was that localized baseline and disease models need to be built in order to predict metabolic risks. So despite this extreme heterogeneity of microbiome composition, certain structural features of the gut ecosystems have shown results that are more consistent across different cohorts. One of these measures is alpha diversity, which, as Christian mentioned, represents within sample diversity. Shannon diversity is one of the most commonly used measure for alpha diversity in the literature. And also, as Christian mentioned, it takes into account both the taxonomic richness of the sample, meaning how many different species are represented, and the evenness, meaning how similar in frequency are the species that are represented in a sample. So Shannon diversity has been suggested as a marker for microbiome health. Lower Shannon diversity relative to healthy has been reported in an individual diagnosed with intractable with IBD, as well as in people with enteric infections such as C. diff, as mentioned by Christian. Increased Shannon diversity has also been reported in hunter-gatherers, and that suggested that either the unprocessed diet, the active lifestyle, or in turn increasing exposure to parasites may contribute to a more diverse gut microbiome. But not only good things are associated with high diversity, as constipation and harder stool are also associated with higher Shannon diversity. This suggests that higher diversity may not be optimal over a certain threshold. So let me tell you a little bit about scientific wellness. Scientific wellness is a rising field, and what it essentially means is that it takes a wellness perspective, which is to try and optimize health when people are still healthy, and to minimize the chance for a disease coming down later in their life, in people's life. The field is based very deeply in science, because in a way that hasn't been possible before, because we didn't have the data tools at the cost that we have today. So the development of new technologies and the large reduction in costs have made it feasible to generate large multi-omic data in the form of what we call a personal dense dynamic data clouds, which we term PD3 clouds. So what is shown in this slide is a study that was done in our group several years ago. In this paper, we perform analysis on 108 individuals, which we call the Pioneer 100. And we utilize data on their genomes, proteomes, metabolomes, and microbiome data. And we correlated the different blood and other measures. And the associations and the structure of the association networks resulted in a number of important biological insights. Through a partnership with between ISB and a spin-out company called Arivel, we have expanded the size of this cohort and added individuals who have consented to allow the de-identified data to be used for research purposes. And the expanded data, which is at the size of about 5,000 individual, we call the Arivel data set. The data available for participants in the Arivel cohort, so as I mentioned, includes microbiome coupled with human genome, metabolome, proteome, a wide panel of clinical labs, as well as data from wearable devices, health questionnaires, and coaching interactions. So this data can be leveraged to provide deeper insights into the biological functions across systems, including the gut microbiome and its relationships to the physiology and host health. The first analysis that we will mention today examines the associations between a subset of these data sets, which is the blood metabolome, gut microbiome, the blood metabolome and gut microbiome alpha diversity. We utilize data from 16S rRNA sequencing, as we discussed today, untargeted metabolomics data measured by metabolome for over 650 blood metabolites, 3 Olink panel, two cardiovascular panels, and 1 inflammation-focused panel, and blood levels of over 260 proteins measured by Olink, as a wide panel of 77 clinical analytes, and self-reported data on gastrointestinal health, disease diagnosis, and dietary questionnaires. So this is our paper. The question is, can we use this deep phenotyping data to understand what is a healthy microbiome? So our work started with the hypothesis that it might be possible to predict gut microbial alpha diversity from analytes that are measured from the blood, and specifically blood metabolites. If successful, this type of approach could be used for early diagnosis of low gut microbiome alpha diversity, which is potentially problematic. As I mentioned, it's associated with multiple conditions. For example, recurrent C. diff infection, as we actually saw in the exercise today. And that condition actually accounts for a large number of deaths in the US. Okay. Yeah. So why did we choose to focus on the blood metabolome? There's a diverse set of compounds that enter our gut lumen through the diet, for example, and they get absorbed into the blood. These are metabolites. Some metabolites come directly from the diet. Some undergo some processing by the digestive system. And another group are what we call microbial metabolites, which are synthesized by the gut microbiome. Some metabolites are subsequently modified by the host, for example, in the liver. And these are called microbial co-metabolites because they are metabolized together by the microbiome and the host. Based on this, we hypothesized that the metabolites present in our blood could reflect the composition of the gut microbiome. In addition, while, as we saw, gut microbiome changes across different environments and geography or diet, we have hypothesized that these small molecular weight compounds may funnel down into a more universal set of blood metabolites that will end up affecting our physiology and will likely be more translatable across cohorts. So in this slide, we can see results from a PNAS paper from 2009 that kind of sets the stage for our analysis. In the bottom, you can see that almost all microbial metabolites are not detected in a germ-free mice compared to control mice, such as indole-3, propionic acid, serotonin, and indoxyl sulfate, but their substrates are detected in the germ-free mice. This suggests that, at least in animals, there is a connection between the microbiome and this small molecular weight compound that we measure from the blood. So in order to investigate the relationship between the host metabolome and gut microbiome diversity, we applied the LASSO machine learning method to baseline plasma metabolomics data from the Airville cohort. LASSO is a computationally efficient penalized regression method, which is very useful for feature selection in data sets such as this, where you have a large number of features, and many of them could actually be collinear and not contribute new information. The inclusion criteria for our discovery cohort required that the initial blood draw was taken within 21 days of the stool sample, because we wanted to prevent a long time passing between when the sample were collected for microbiome analysis and for the blood metabolomics. So we also required that the microbiome was measured by DNA Genotech, which is one of the vendors in the Airville cohort, and that their clinical labs were analyzed by LabCorp. This kind of narrowed down in our cohort to a size of 399 participants. So to predict gut microbiome alpha diversity from each of the different omics, and specifically from metabolomics, we use a scheme of 10-fold cross-validation, which basically means that we randomly partitioned the data set into 10 groups of participants, and we then trained the model each time on 90% of the data using internal 10-fold cross-validation, and then use the model to predict the remaining 10% who were not used to train the model. So this was repeated 10 times for each of the partitions, and the end result was 10 different models and 10 different beta coefficients for each metabolite, and 10 different out-of-sample predictions of Shannon diversity for each participant. This schema generally minimizes the chance for overfitting. And we evaluated the accuracy of the model using out-of-sample predictions. Okay, so a little bit about our cohort. The participants in Airville are generally healthier than the general population. They're still very reflective of the states people came from, which is mainly Washington and California. Nevertheless, we still have a significant representation of all classes of obese individuals. We also noticed that we chose to stratify people's BMI based on the WHO definitions, which is obese 1, 2, and 3. The importance of this refined stratification will become apparent later. As expected, markers of health such as CRP and triglycerides are decreasing with increasing BMI. Okay. So to our surprise, we found that not only were we able to predict gut microbiome diversity from 659 metabolites measured for each participant, but we could also do it using a small subset of only 40 metabolites that were retained by at least one of the 10 LASSO models that were used to predict Shannon diversity in our process. So the upper plot on the right represents the presents the metabolome predicted Shannon diversity, which we call M Shannon on the x-axis, versus the observed Shannon diversity on the y-axis. So looking at this plot, we see that this small subset of 40 plasma metabolites was strongly predictive of Shannon diversity and was able to explain 45% of the variance in Shannon diversity with a Pearson of 0.68. The bottom right plot shows the average out of sample R square scores from models predicting different measures of alpha diversity, including Shannon, PD whole tree, and Chow1 diversity, using either only the 40 metabolites that were retained in the model predicting Shannon diversity, and this is shown in red, or the whole metabolome that is shown in gray. So we see here that the LASSO model fitted using tenfold cross-validation with only 40 identified metabolites did a pretty good job also explaining the alpha diversity, the other types of alpha diversity, 50% for PD whole tree and 36% for Chow1. We also see that using the whole plasma metabolome did not significantly improve the performance, the predictive capacity of the model. And that confirms that the small subset of metabolite that we identified is sufficient to capture the majority of the explainable variation in Shannon diversity by blood metabolome. When we further narrowed down the set of metabolites to only those who were retained by all of the 10 LASSO models built in our process, the intersecting set included 11 metabolites, which we call core metabolites. When we use these 11 metabolites to classify participant with low alpha diversity, meaning in the bottom, which we defined by alpha diversity in the bottom quartile for our cohort, using a cross-validation implementation of random forest. So the 11 metabolites alone were able to classify participants on the basis of the 3 alpha diversity measures that I mentioned, with considerably high sensitivity, specificity, and precision. These results indicate the potential to use these 11 metabolites as biomarkers for gut microbial diversity. So let's look a little bit on the core metabolites. So nearly a third of the metabolites, actually on all the 40 metabolites, so a third of the 40 metabolites identified in our analysis were previously shown to be synthesized by gut microbiota. Another 9 are potentially microbial. These are again are microbial metabolites are again those that are either initially synthesized by the host and then further metabolized by the microbiome, for example, bile acids, or the other way around, first synthesized by the microbiome and then modified by the host, for example, hippurate. So our results confirmed metabolites that were previously tied to alpha diversity, such as hippurate and p-cresyl sulfate, and identified new candidates biomarkers for Shannon diversity. Would specifically like to mention the top one. This is the highest positive metabolites that had the highest average beta diversity across the 10 model is 5 alpha androsten 3 beta 17 alpha disulfide. And this is a testosterone metabolite. We found very little information about it in the literature. And it was higher in men versus women, while Shannon diversity itself is not significantly different between males and female, even when you adjust for age and BMI. These 2 secondary bile acids two secondary bile acids were also retained in all the 10 LASSO models, but they demonstrated opposite associations with Shannon diversity. Several of these microbial metabolites that were retained in our LASSO models were shown to to exert biological effects on various organs, such as the kidney, liver, and the heart. And I'll give you a few examples. Hippurate is a microbial metabolite which is formed in the liver from benzoate, and it belongs to a diverse group of phytochemicals, compounds that are called polyphenols. They are found in fruits, vegetables, cereals, coffee, tea, and red wine. The positive association between polyphenolic metabolites and Shannon diversity suggests that a diverse microbiome may reflect polyphenol-rich diet. Hippurate was also associated with improved metabolic function in the twins UK cohort. Another example is the microbial metabolite imidazole propionate, which was negatively associated with the Shannon diversity in our models. A recent study demonstrated that imidazole propionate is synthesized in greater abundance in the gut microbiome of diabetic patients, and that it is able to impair insulin signaling in animal models, directly working on the liver and pancreas. Both of those examples support the notion that higher microbial alpha diversity is associated with better health. However, our results also suggest the opposite. For example, p-cresol sulfate was positively correlated with Shannon diversity, but this compound is a potentially toxic uremic compound. Similarly, TMAO was positively associated with Shannon diversity, independent of age, sex, and BMI. And TMAO is associated with diet high in red meat and was previously suggested to be associated with cardiovascular disease. So I'll give Tom the floor.
Thanks, Noah. So I'll take it over from here. So once we discovered the array of metabolites that show the strong correspondence to alpha diversity, we want to take it a step further and see the known gastrointestinal disorders and certain lifestyle habits that are associated with lower alpha diversity. Is the reflection of these metabolites consistent across different disease states? And to this end, we regressed both actual measured Shannon diversity and in separate models, the metabolome prediction of Shannon diversity against different diseases seen on the slide here, adjusting for age, sex, and BMI. As you can see, for example, abdominal pain or type 2 diabetes is associated with a decrease in Shannon diversity. But quite interestingly, the metabolome predictions in almost every case move the same direction. Similarly with diarrhea, which is also shown in the box plots to the right, with higher frequency of diarrhea, you have a decrease in alpha diversity. And this is quite strongly reflected in the metabolites we see in our metabolomic prediction, showing that different lifestyle factors and diseases can impact your gut microbiome, and the reflection of the metabolites consistently shifts in the same direction. We next want to ask an important question is whether antibiotics use disrupts the accuracy of our models. Antibiotics are known to disrupt gut microbiome. They're known to decrease alpha diversity, but also they can impact the gut microbiome in various different ways depending on their mechanism of action. So in this case, if we remove antibiotics users from our models, does our prediction actually increase because we eliminate some of the noise? To our surprise, and shown in the figure to the left, whether we just performed the analysis on confirmed non-antibiotics users or the whole cohort, our prediction accuracy was very, very consistent, meaning that antibiotics do perturb the gut microbiome, but our metabolic predictions are quite good at capturing that perturbation. We also looked at the time lag, since remember, if you recall, we specified that individuals had to have their blood taken within 21 days of their stool sample. But we wanted to see that whether if you're closer to your blood draw, are the models better or worse? And what we observed is that the models don't get any better if you get very close to the blood draw and your stool sample, but there is a decrease in performance the further away you are. So in tertile 3 of our participants, so past 10 days, we still get a pretty good performance, but there's a significant drop. So there really is this 10 to 14-day window where we get best performance, where the metabolites correspond to your gut microbiome the strongest, which is great. And it can also lead to future diagnostic recommendations and how these metabolites can be used in a clinical setting. We next wanted to compare how these metabolites perform relative to what's really the golden standard in the clinic in evaluating people's health at a molecular level, which are standard clinical lab tests. And these include triglycerides, cholesterol measurements, CRP. To this end, we stratified the cohort into quartiles based on alpha diversity. And we want to predict or identify or classify individuals in the lowest quartile of SHAN diversity versus the whole cohort. And quite surprisingly, the 11 core metabolites Noah mentioned were outperformed a whole panel of clinical labs to a pretty large extent, showing that these metabolites are quite robust predictors of and can be potentially used to identify people at certain risks because of their low alpha diversity. But also, I think more importantly, we demonstrate that things we measure in the lab and all the clinical lab panels you get tested at in a doctor's office don't very strongly reflect the health of your gut microbiome, and potentially other tests are needed to do so. We next actually took the Arvil cohort to our advantage since as Noah mentioned, there are actually 2 different microbiome vendors used throughout the course of Arvil and its existence as a company. So we took our models and wanted to see how well did they perform if we use a completely different gut microbiome vendor with a different processing pipeline, different library. And the results were quite consistent. As you can see to the graph to the left, whether we just used our 4D metabolite panel or the whole metabolome, we still could predict a big amount of variance in between people in alpha diversity. Consistent with our random forest analysis from the slide previously, clinical labs showed very little signal when we performed the same analysis. And similarly, a panel of over 200 different proteins showed very little correspondence to gut alpha diversity, really pinpointing the metabolome as this strong reflection of what's going on in our gut microbiome. And when we fitted the models independently using cross-validation, which is shown on the plot to the right, the coefficients within the models for the 11 core metabolites were very consistent between the 2 groups. So as you can see, isouracil deoxycholate, for example, was a strong negative predictor in both the models trained on the validation cohort and the discovery cohort, showing further the robustness of our findings. So what insight do we get from microbiome health and the role of alpha diversity as a metric of microbiome health? As Noah pointed earlier, several of the metabolites negatively associate with alpha diversity. So metabolites that go up when alpha diversity goes down, like imidazole propionate, are actually detrimental to human health. They can directly promote insulin resistance, indicating that low alpha diversity, consistent with previous studies, is probably not optimal for human health. However, there might be an upper threshold beyond which alpha diversity, too high alpha diversity, can actually be detrimental to human health as well. And that's because of the concentrations of different uremic and cardiac toxins that rise with the diversity of your gut microbiome. TMAO being one example, p-cresol sulfate another. So there likely exists this Goldilocks zone where too much is bad and too little is too bad. And if we can get people in this optimal range, that could be ideal for their health. What's worth adding as well is that this ideal zone may vary depending on each individual's health state and physiology, age even. People who, for example, have compromised kidney function may actually benefit more from a slightly lower alpha diversity due to their inability to excrete these uremic toxins through the kidneys, which is the main route of exit from the system. So having developed these models and really diving into SHAN diversity in the Arvil cohort, we wanted to tackle a certain inconsistency in the field, which is the association of alpha diversity with obesity. Previous studies were quite inconsistent in whether alpha diversity goes up or down or remains the same in obese people versus normal weight individuals. To shed some light on this, we actually divided our cohort not just into obese and normal weight categories, we also extended it to the World Health Organization categories of obese. So obese 2 and 3 are individuals whose BMI is over 35, so what could be considered morbid obesity. As you can see in this graph, that is the group that showed the strongest decrease in alpha diversity relative to normal weight individuals. While obese 1 individual still had a slightly decreased alpha diversity, it was less pronounced than in that obese 2-3 group. Consistent with this amplified negative effect in obese 2-3 individuals, they also demonstrate a much higher level of C-reactive protein, so higher levels of inflammation in the obese 2-3 group relative to the obese 1 and normal weight individuals. And that is shown in the box plot in the top left of the slide. But what we really were curious in asking is whether the metabolites we identified, does their association with gut microbiome health and gut microbiome structure, is it altered in extreme obesity? So to this end, we took our 5 strongest predictors from our analysis and regressed them against alpha diversity, adjusting for covariates in each of the obesity groups separately. And while most of them show very consistent associations with alpha diversity, our main predictor, 5-alpha androsten 3-beta 17-alpha, showed virtually no association in obese 2-3 individuals, while a strong association in all other people in our study. When we extended that analysis to all 11 of our core metabolites, other differences emerged. One being PFAS, which is actually a global pollutant, showed a positive association with alpha diversity in obese 2-3 individuals, while no association in normal weight individuals. Once again, pointing that there's a perturbation in the blood metabolome gut microbiome relationship in extreme obesity that's worth pursuing further. So to summarize the results of this first project, we were able to very accurately predict alpha diversity using just 4 of the plasma metabolites, but clinical lab tests and proteomics could not perform the same way. Metabolomics were by far the strongest signal. Specific metabolites associated with alpha strand diversity were also related to human health. And probably what's most striking is that there was a variable relationship between our specific metabolites and strand diversity across the BMI spectrum, with particular disruption to this relationship in severe class 2-3 obesity. The question that really remains and that we have some theories about is, why is this possible? Why such a small subset of metabolites measured in the blood can capture such diversity in gut microbiome structure across individuals? And for that, I think we have to go back to some basic nutrition and understand diet and really the components we consume in a regular diet. The macronutrients and micronutrients that we focus on most, which are fats, carbohydrates, essential vitamins, amino acids, they actually comprise a very small part of the diversity of our diets. What really separates us from others is this wide array of phytochemical plant compounds that we consume when we drink coffee, when we eat certain vegetables. Over 8,000 structurally different phytochemicals have been identified, and over 1,000 different foods compounds have been identified in foods. And these compounds are not absorbed in the small intestine, so oftentimes they end up in the large intestine in millimolar concentrations and directly interact with our gut microbiome. This diversity of phytochemical compounds that makes it into our large intestine is not only metabolized by the gut microbiota, but potentially influences the gut microbes present, since many of these polyphenols and phytochemicals have antimicrobial properties. So it contributes to shaping the gut microbiome and likely probably explains some of the vast inter-individual variability between people. But despite their structural diversity, they're all broken down to a much narrower, smaller group of universal compounds, usually phenolic acids, hydroxyphenolic acids. And those were some of the strongest predictors in our models, suggesting that there's this funneling effect of very diverse diets, very diverse microbiomes into a smaller subset of metabolites we can quantify. And this can lead to a more universal measure of gut microbiome health moving forward. So that completes our first project. And we wanted to present some of our work that's ongoing, that's on bioRxiv right now and in review. And that's utilizing deep phenotyping in large cohorts to understand certain patterns in the gut microbiome across aging and healthy aging. And this preprint is on bioRxiv, so you can find it there, and hopefully will be published soon as well. So a bit of background about what we know so far about aging in the gut microbiome. There's been plenty of research done in early stages of development in children, and quite considerable literature as well in old age. And the picture that it collectively paints is that in the first few years of our life, from between 0 to 3 years of age, there's high instability in the gut microbiome. Upon birth and breastfeeding, transitioning to solid foods, there's this heterogeneity in your gut microbiome composition. Around 3 years of age, it stabilizes, and you slowly enter this adult-like microbiome, oftentimes dominated by Bacteroidetes and Firmicutes. And this microbiome that you have as an adult is thought to be relatively stable up until old age, about 65 plus years old, at which point gradual compositional changes occur. And these often correspond to decreased alpha diversity, Oftentimes, frail individuals who enter long-term care have this decline in diversity-associated taxa and short-chain fatty acid-producing taxa. And usually this is associated with various measures of age-associated decline. However, there is this inconsistency in the field because several studies have been conducted where gut microbiomes were sequenced of extremely long-lived individuals. These are people who live over 100 years and are functional, are healthy, still frail, but have lived a very healthy and long life. And it oftentimes, in this cases, we see a very different signature than what happens in old age and what happens in adulthood. Oftentimes, alpha diversity is increased. There's a decline in core taxa, or core microbes that are shared among individuals, an increase in various rare microbes, both beneficial and pathogenic at the same time. But because of the small sizes of cohorts involved in longevity research, our understanding of this is very minimal since there's not that many long-lived individuals who provided stool samples. But using deep phenotype cohorts can give us some insight in actually these aging trajectories and whether what we see in centenarians happens decades earlier in life. So to this end, we use once again the Arville cohort, this time combining both vendors and using all gut microbiome samples, not just the ones that had paired metabolomic data. And we also collaborated with Eric Alwall and the Mr. Oz Consortium, which is a cohort of older men originally recruited when they were 65 and over. A prospective cohort study trying to evaluate risk of fractures and osteoporosis in men prospectively. At their fourth follow-up visit, when all the individuals were over 78 years old, a subset of them provided stool samples, and we actually had follow-up data on their survival as well, which we used in this analysis. So just to briefly, once again, an overview of what happens in extreme longevity. There's this depletion in core taxa, and we see this rise in certain rare microbes, which has been previously termed as this acquisition of a longevity-adapted subdominant fraction. And these are both pathogenic and beneficial microbes. But we have this idea that potentially this isn't necessarily detrimental or a burden on the host. It actually may be the gut microbiome's adaptation to the changing physiological landscape with aging. And this adaptation can actually be beneficial and even contribute to longevity. But how do you capture this in a large, diverse cohort? And how can we measure whether this pattern originates earlier or at the very end stages of life when you're close to approaching longevity? Our approach was to really focus on this rare fraction. We had this idea that if this pattern occurs earlier, it should probably be characterized by this decrease in core taxa, a rise in rare taxa. But we further hypothesized that the gut microbiome may actually become more personalized. These rare microbes rise, but the microbe that rises in each individual may actually be very specific to that person. So we took a beta diversity approach to this question, where you calculated the similarity matrix where each sample is compared to everyone else in the cohort. We then take the minimum value for this calculation, which really says how close are you to your nearest neighbor in a large reference pool of thousands of individuals. And then we take that value and then we use it for downstream analysis. As you can see, both at the genus and ISB level, this metric is normally distributed in a population, which makes it quite good for statistical analysis downstream. So quite interestingly, we observed that both at the ISB and at the genus level, gut microbiomes become increasingly unique as you age in the Arville cohort. And the first signs of this increased uniqueness occur at around 40 years of age at the genus level and around 50 years of age at the ISB level. And these are models adjusted for BMI, sex, alpha diversity. We further even validated the same pattern in a separate cohort of over 4,500 individuals, mostly from UK and the US, the American Gut Project, where we saw very, very similar patterns, the same increase with age and uniqueness, particularly in 80-plus-year-olds, showing that this metric or this trend we're seeing is quite consistent across very different cohorts. It happens over and over again. To gain some insight into what actually, if there's this divergence in microbiomes, is there any convergence in phenotype? And because of our previous work with Noah showing the strong correspondence with the plasma metabolome and the gut microbiome, we once again dove into the metabolomics data for Arville participants and regressed how unique you are to each metabolite, adjusting for, once again, alpha diversity, sex, and the other covariates you see in the bottom of the slide. To our surprise, we had about 8 or 9 hits from the panel of 650 metabolites. All of them were microbial in origin, which already was quite exciting, that they're not only strongly associated with uniqueness, they actually are derived from the gut microbiome. Interestingly, some of them, for example, the strongest metabolite associated with our uniqueness measure, phenylacetylglutamine, has previously been proposed as a healthy aging longevity marker, since Italian centenarians show consistently higher levels of phenylacetylglutamine relative to elderly and young controls. The other analytes that really emerged from this analysis were also indoles, which are tryptophan degradation products in the gut microbiome. And this is particularly interesting because these indoles have a very known immune modulatory role. Different bacteria break down tryptophan into a variety of metabolites, including indole propionate, indole 3-acetate, and indole itself. These metabolites are absorbed by the host, and that's why we can measure them in the plasma. But they also act on very specific receptors, one of them being the aryl hydrocarbon receptor, AHR. And in several animal studies, feeding mice, for example, indoles, was able to extend their health span and survival through action of that receptor because of its modulation of the immune system. So this kind of points us that this trajectory we're seeing might actually be reflective of healthy aging. If these metabolites we're seeing are changing in a way that's consistent, where immune function may be modulated, where there's this adaptation to the host aging, this could actually mean that this is favorable and something that's desirable and should be pursued. But to really validate that, we need to look at the latest stages of life, where individuals are close to approaching longevity, but also where there's more health heterogeneity, where people have over time either acquired certain chronic conditions and diseases or have remained healthy throughout their long lifespan. Hence, we return to the Mr. Oz cohort, which at the time of sampling of the gut microbiome, all individuals were over 78 years of age. The oldest individual was 98, and the mean age was 84. And here are some demographic characteristics stratified by medication use. As you can see, there's quite high prevalence of diabetes, of hypertension, particularly in the high med group. So we have this uniqueness pattern identified in the gut microbiome, and now we want to see whether it's reflective of healthy aging. Our idea was to look at various measures of health in older individuals, but health and healthy aging is not an easy metric to capture. There's self-perceived health, there's actual functional measures, there's comorbidities. So we focus on 4 different metrics. One is medication use. The more medications you have generally reflects more comorbidities, more diseases. But we compare that with self-perceived health, like whether you think your health is excellent relative to your peers, or you don't think it's as good. Life space score, which actually tells you how functional you are. And walking speed, which is arguably the most validated measure in older populations that reflects your functional status, is indicative of survival, of executive function, with faster walking speed meaning you're healthier, and slower walking speed meaning you're less healthy. And finally, we created a composite healthy score, where an individual had to be healthy in at least 3 or more of these criteria to meet our healthy composite definition. Quite strikingly, under every stratification we performed, the healthy groups showed this consistent pattern of increasing uniqueness with age. And that's shown by the Pearson correlation coefficients for each group with uniqueness on the far left. In the less healthy group, in every case, there was virtually no association. It was pretty much a flat line. To further confirm this isn't some artifact or some reflection of alpha diversity, we actually compared it to different alpha diversity measures. But in each case, the trends were not there. Healthy individuals did not show a rise in alpha diversity. It was pretty much no signal across both groups studied, indicating that this uniqueness signal captures something independent of just microbiome richness. We paired our uniqueness analysis with differential abundance analysis of microbes that consistently either rise or fall with healthy aging in the healthy group, and separately in the rest of the cohort that did not meet our stringent criteria of what we define healthy. As you can see, the only genus that emerged as a significant hit was Bacteroides, which is one of the most dominant genera in the human gut microbiome. As you can see, as healthy individuals age, this core microbe actually gradually decreases, and you just don't see the same pattern in less healthy individuals. When we then looked at specifically 85-plus-year-olds, so the oldest people in our cohort, and we looked at their survival 4 years in advance as they're approaching extreme old age, we observed that both relative Bacteroides abundance and our uniqueness measure associate with survival. So the higher your Bacteroides, if you retain Bacteroides into extreme age, you're less likely to survive in the course of our follow-up. Similarly, if your uniqueness score is lower, you're more likely to die than someone who kind of went on this uniqueness trajectory and is microbiome adapted to their aging phenotype. So in conclusion, Noah and I think both hopefully show that deeply phenotyped cohorts can provide pretty novel and interesting insight into gut microbiome health. And using some of these higher level gut microbiome measures like alpha diversity or beta diversity can allow us to find more consistent associations between gut microbiome and host physiology. And hopefully, as we've shown in a few instances here, these analyses and these comparisons and associations actually do quite well validating across very different cohorts, very different processing pipelines, very different libraries. However, I think there's still a lot of validation to be done. For our metabolomics and alpha diversity paper, I think a major validation is that does this pattern hold across vastly diverse cohorts? If we measure this in the Asian cohort or a European cohort, do the same metabolites correspond to alpha diversity? How universal is this signal? Some preliminary evidence that came out this year from the Twins UK cohort and another Danish European cohort suggests that these metabolites actually are indeed, at least in the European population, quite consistently associated with alpha diversity. But now that we know this association, how can we actually personalize interventions to either change alpha diversity or these specific plasma metabolites? For example, we may want to decrease TMAO in an individual and imidazole propionate, but they might actually benefit from a higher concentration of indoles or indole propionate. And certain methods like Christian presented with MyCom can actually get us closer to this goal, where we can engineer the gut microbiome through these more complex modeling techniques to get at the desired outcome. And I think a big question with our aging analysis is, does the gut microbiome simply reflect healthy aging? Or does it actually promote it through the synthesis of the metabolites we discussed, primarily tryptophan and indole metabolites highlighted in our analysis? So a lot of questions remain, but very exciting, and hopefully we can answer some of them ourselves. With that, I'd like to acknowledge our whole research group and everyone who helped on the projects, and our collaborators, including Sean Gibbons, who was crucial in both of the projects presented, Christian Diener, who was absolutely phenomenal and helped a lot in the aging project in particular. My PIs, Nathan Price and Lee Hood, as well as our collaborator, Eric Orwell from OHSU. I think now we're gonna have a 3-minute break for everyone to get water or have a bathroom break, and then we'll have closing remarks by my boss, Nathan. Thank you, everyone.
Okay, I'm on. Looks like I'm on. All right, well, welcome everyone. Hello, I'm Nathan Price. I'm an associate director and a professor here at the Institute for Systems Biology. What a wonderful day we've had. I just want to thank all the people that have been involved, and especially first our sponsors, Metabolon and the Washington Research Foundation. We couldn't do this program without you, so thank you so much for the support. I want to thank also the team here at ISB for making this event happen. First and foremost, our faculty lead, Sean Gibbons, who's been tremendous and has brought wonderful microbiome expertise to ISB, and we've been so pleased to have him here for the last little while. Huge thanks also to our course facilitator, Christian Diener, who has driven so much of the work that you saw today. I just want to thank him for his contributions in putting this course together and for driving forward so much of what's happening here at ISB. I want to thank the teaching assistants, Tom Womansky, Noah Rappaport, Alex Carr, and Priyanka Baloney, who have, again, are the core of our microbiome team here, and I think did a great job in doing their presentations today. I also want to thank the administrative team working behind the scenes here, Joey Petoza, Allison Kudla, Joe Mixter, Audrey Hubbard, Katie Keppert, Renee Duprell, and Thea Swanson, who did all the work to make this go, I think, pretty reasonably smoothly. So we're excited about that. So as a nonprofit organization, we also rely on the generous support of our donors. ISB has pivoted quickly in the wake of the pandemic to put on a number of virtual events such as this one at no cost to registrants. We would greatly appreciate your support, which is needed now more than ever. So if you're in a position to do so, we'd please consider making an online donation at isbscience.org/donate. Very much appreciate that. So I'll just briefly recap what we learned today, not in detail, it's the end of a long day, but hopefully you were able to learn a lot about at least the way that we work on and view microbiome research here at ISB, starting with the great presentation this morning that Christian gave, going through how we think about this from a sequence point of view. Later on, also looking at how we can think about functional analysis of the microbiome through metabolic analyses, MyCom being one of the examples that we went through. And then what you saw then just more recently from Tom and Noah really gets into the issues of how we think that the reflections in the blood, in the metabolites, actually from different tissues in the human body or through a host lens, really give us a way to understand functionally what's happening in the microbiome. And so you've heard, I won't rehash all of that, but those things together, along with the presentations from Alex and Priyanka as well, we think those together give a view of at least the way we're thinking about microbiome. We look forward to hosting you here again tomorrow for our symposium. It's going to feature, here we're going to be moving outside of just our group here presenting to give a broader view of what people around the world are doing on the microbiome. So it's going to feature 6 researchers working in different parts of the world to build computational tools for manipulating the gut microbiome to track and treat disease. We'll kick things off tomorrow at 9 a.m. Pacific time. I won't even try to translate that to all the time zones that are represented here in the audience, but it will be 9 a.m. Pacific time, which is what it is here in Seattle. And we hope to see you here then. Please learn more about ISB at our website, isbscience.org. Thank you so much to everyone for joining us, and have a great day.